Good morning, everyone. We're going to go ahead and get started. We're very excited to have you here in person for our Interact Symposium today. It's indeed our pleasure to welcome you both in person as well as virtually. We have a number of international guests that are joining us virtually, and so welcome at least electronically to Stillwater, Oklahoma. So we're very privileged to be in the McKnight Center today, and we have an outstanding group of speakers to visit with us about research and focused on the research that we're looking at for Interact, which of course is the Institute for Translational and Emerging Research in Advanced Comparative Therapy. I want to recognize a few of our VIPs that are here today with us. Of course, we have our Nobel Laureate. Dr. Beutler is here, and we're excited to hear him later on this morning, as well as Dean Risco from the College of Veterinary Medicine and Dean Krutz from the College of Arts and Sciences. We will have others joining us in and out today, but without further ado, I think we'll go ahead and get started. Again, these are our Interact committee members. We encourage you, if you did not pick up a compendium when you checked in this morning, to grab one of those so that you can have those abstracts. Just a few kind of housekeeping announcements. We do ask that masks be worn, as OSU expects. We do ask that you maintain social distancing, and if you are to be coming down front, we do have a microphone to ask questions from our live audience. It's down here just in front of me. Watch that step there, so don't join us in the first 3 rows because there is a step there. Cell phones, we ask that you take those electronic devices and make sure they are either on silent or vibrate if you've not already done that this morning. A few notes for our presenters. Be sure that you checked in with us so we know that you're here, and please be backstage to my right at least 10 minutes prior to your presentation. Our poster presentations can be found in the East Lobby, and they will be running all day. We do ask that our poster presenters be there at 1 o'clock to address any questions by our judges. We'll also have a photo session this afternoon for our presenters at 2:15, and that will be out in the lobby as well, so be listening for additional announcements. And of course, join us for a bit of celebration this afternoon at 4 o'clock for our awards ceremonies to recognize our award winners of both the in-person and the poster presentations. So our first presenter this morning is a student from the College of Engineering, Architecture, and Technology here at Oklahoma State University, Yu-Xuan Li, presenting on a multi-layer network-enabled ultrasonic image series analysis approach for online cancer drug delivery monitoring. If you'd like that.
Oh, right.
Okay, or you can use the mouse pad. Okay.
All right. So this one will just.
Mm-hmm.
Oh, okay. All right.
Okay.
Thank you.
All right.
So, good morning, everyone. My name is Yu-Xuan Li, and I'm a PhD student from School of Industrial Engineering and Management, Oklahoma State University. I'm working with my advisor, Dr. Chen Liu, in STARS Laboratory now. And it's my honor to present my topic here. And today, my topic is a multi-layer network-enabled ultrasonic video analysis approach for online cancer drug delivery monitoring. This work is also collaborated with Dr. Rajan in NTTL's laboratory. So I will first introduce the problem, and then I will talk about my proposed methodology and validate my methodology based on 2 case studies. One is simulation, another one is a real-world case study. And then I will talk about what we are doing right now, and then I will give a summary. So let's start. In the healthcare systems, the medical images becomes a very important tool to help the doctors to do the diagnose. Among all the imaging techniques, ultrasound image is a very important one because it's cheap and it's safe and it's easy to get and it's simple. And based on the previous works from Dr. Rajan's work, they have shown that we can use the ultrasound image to do the image-guided drug delivery to monitor the drug delivery process. However, because of the high noise in the ultrasound image, it will make the analysis challenging. And in order to address these gaps, we developed a new method for capture the changes of ultrasound images and then monitor and evaluate the whole drug delivery process based on the proposed method. So this methodology contains 5 steps, and it involves, first we will do the ROI identification, and then we will partition the image to transform it as a multi-layer network. And we will explain why we use that in the following slides. And then we will use the community detection to apply the multi-layer network, and then we will extract the features for monitoring the drug delivery process. I would like to first introduce the definition of the multi-layer network. You can see in the figure 3. Oh, sure. Oh, this one doesn't work. Oh, here. So you can see that we have several different layers. And for each layer, we will have the same node, 1, 2, 3, 4. But for the different layers, the link weight between different nodes are different. So we can consider it in the image way. We can consider each layer as the ultrasonic image. And then the link weight can be considered as the information from the ultrasound image. And then the difference between the layers can be considered as the difference between the ultrasound images. And then we combine these layers together. This can be a multi-layer network, and this one can also represent the whole ultrasound image series. And we can consider in this way. We first do the image partitioning, because we want to make sure that for all the images, we have the same number of nodes. And then we will calculate the spatial regularized distance. We can get the adjacency matrix of the whole graph. And here provides an example of the demonstration from the image to the multi-layer network. So you can see that we first use the same way to participate the images, and these four images from image 1 to image 4, they are the images from the same ultrasonic image series, but they will have slight difference, but it's hard to see. And that's the reason why we need to transform this into the multi-layer network. For each image, we'll transform it into one layer. Image 1 to layer 1, image 2, layer 2. And you can see that the differences between the images can be transformed as the link weights between the different layers. And then we can combine, Then we can combine the 4 layers together as a multi-layer network. And we can use the multi-layer network to do the following analysis. So next part is to calculate the spatial regularized distance, and that is the part we need to calculate the link weight. So we have provided 2 parts. First one is the node similarity, another one is the spatial distance. So we provide an example here. You can see node 1 and node 2. And they have the similar value of the vector, so they have a very high node similarity. And also, you can see that they are separated only by 2 units. So if we calculate the Manhattan distance, the spatial distance is also low. So the high node similarity and low spatial distance will give us a high link weight, and we can calculate all the link weights between each 2 nodes in this way. Then we can get this multi-layer network, and then we can do the community detection. So the community detection follows the Debacle's work, and we can identify n groups in one multi-layer network. And figure 7 shows an example. That's when n is equal to 2. And you can see that assume the yellow area, that is the ROI, and we can get 2 different communities. One is still the yellow area, another one is the blue area. And you consider if we have several multi-layer networks, Then if we consider the changes from each network, from the first multilayer network to the second one and to the third one, and then we can visualize the changes as the drug delivery process. And after the community detection, we would like to do the feature extraction. So our proposed method just calculates the pixel intensity of the area we have extracted or detected. And then we also put another conventional method here, and this method is proposed by Dr. Rajan's work. It just calculates the intensity sum of the pixels of the whole area. And then we would like to do the comparison to show the effectiveness of the proposed method. And the metric here is used as the feature increment percentage, and I will explain it in the case study. First, we use a simulation to demonstrate the effectiveness, and we first generate the ground truth in figure 8a. And you can see that the inner square is increasing, and the cell length is increasing linearly. And then we can transform it to the ultrasonic image series based on the image simulator. And we also labeled the area that is splatter, that is surrounded by the red rectangle. And we can use this image series for the following validation. And here is the result. You can see that first here, the area surrounded by the red square, that is the actual splatter area. But the area that is blue means the area we detected as the new community. And you can see that the area that we detected and the area that is true splatter area are very similar. So that shows that our method can detect the community effectively. And this part is to show the visualization of the drug delivery process. And then we consider the features. And here you can see that we calculate the features from 4 images, A, B, C, D. And we consider the features from A as the base and calculate how much the features of B, C, D has increased. And that's the reason why we call it as the feature increment percentage. And we put 3 parts here. First is the proposed method. Next one is the size of detected community, which is also extracted by the proposed method, just the size of the blue area, and then the conventional method. And you can see that the pattern of the proposed method is consistent with the conventional method, and is more sensitive than the conventional method. Besides, the proposed method is much similar as the size of the detected community, and this also proves that we have extracted the true area, or means the area that is splatter. After that, we would like to do the real-world study. I would like to first introduce the data, and this data is collected after the mouse is injected with the ELTSL. And it involves 3 different series, and they are collected under different temperatures, 37, 39.5, and 42. We use different temperatures is because of the nantitypos is sensitive to the temperature. So that's the reason why we want to do a comparison in this way. And here, figure 11a shows an example of the collected ultrasonic image. And we do not consider the whole image. We just expand the area here out as the area that we want to consider. And here, the yellow area, that is the ROI. And we consider 2 different methods. First is the conventional method, CDE, and another one is the proposed method, that is FGH. And the images for the same column, they are collected under the same one. So that means the images that are used for CNF are the same image, and the images used for DNG, they are the same image, and the images for ENH, they are the same image. And you can see that the proposed method is also consistent with the conventional method, but it's more sensitive. We also can show this result in the feature extraction. So here shows the features, and it also uses the same metric, that is the feature increment percentage. And you can see that the proposed method has the same trend, but it's more sensitive than the conventional method. And the improvement can even more than 0.1 0.1. So that means we can improve more than 10%. Almost, also we will get the maximum one that is 18%. So this shows the effectiveness of proposed method, and then I would like to talk about what we are doing right now. So we consider 4 aspects for the following analysis. First is the tumor size-based analysis, because you can see that we have 3 different series. That's from the different temperature. But you can see that the ROI of the different temperature is not the same. So we want to compare them. So we need to find a general way to quantify the feature changes. And then we would like to utilize more mouse and treatments, because the images we use now is just from the same tumor, from the same mouse model. So we can consider more mouse model to get an average result, and then we can consider some other treatments for the comparison. And then we are considering the time-based comparisons, because after the mouse is injected with the tumor, the mouse will inject with the treatment on both day 3 and day 6. So we would like to consider the drug delivery process regarding the growth of the tumor. And the last aspect is that we are applying more specific features, because now we just use the intensity value to do the feature extraction, but we consider more features to do the last step. So in the summary, we use the multi-layer network to represent the ultrasonic image series, and then we can apply the ultrasonic multi-layer network to detect the communities and extract the features. And in the future, we would like to extend our analysis from the 2D image to the 3D image, and we can consider the data from other animals and take more nanoparticles into the consideration. And that's it. Thank you so much. So any questions? I will be happy to answer all your questions.
As a reminder, any questions, you're welcome to come to the front of near the stage and ask a question. As our question comes towards the front, I want to remind our online audience we are having a bit of a technology issue. Your slides are just a little bit slanted. We recognize that. We are working on the issue. We do recommend that you view that in full screen view, because that will eliminate it slightly. But we recognize the problem. We appreciate the feedback, and we will be trying to address that throughout the day.
Thank you.
So I have a question about the temperatures.
Yeah.
So in normal physiological conditions, do the temperature around tumors change? And if that's the case, how are you going to use that in order for imaging?
So I think mainly the temperature will change the drug delivery process, not the tumor. So we apply the different temperatures because the ALT/SL, that is sensitive to the temperature. So we would like to do the comparison and to show the effectiveness of this drug treatment under different temperatures. And that's the reason why we have these 3.
So in that case, during the delivery, your proposal is to that particular localized region to have increase the temperature. That's what you're...
Pardon?
In terms of delivery,
Yeah.
so if these tumors are on surface, you can change the temperature to increase the drug delivery. But if the tumors are not accessible, let's say, internalized, so then how are you going to use this to have an efficient delivery?
Oh, so that's a good question. And I think we mostly consider the influence of the temperature for the drug delivery treatment, but we do not consider too much about that. So maybe we can consider later in the future work. Thank you.
All right, I think we'll conclude with questions there. We want to thank our first presenter for kicking us off this morning with a great, great presentation. Give us just a moment. We'll change slides.
Okay, thank you.
Our next presenter is Dr. Yu Feng, Assistant Professor from the College of Engineering, Architecture, and Technology here at Oklahoma State University. And the presentation title is Developing the Next Generation Elastic Whole Lung Model Using Computational Fluid Particle Dynamics.
All right, thank you very much. So good morning, everybody. So it's my great pleasure to have this chance to share our lab's work on developing the next generation elastic whole lung model using computational fluid particle dynamics. So we will briefly introduce a little bit about the concept of in silico personalized pulmonary healthcare, and then we will briefly discuss about how this computational lung model can help on pulmonary healthcare. And then it's about the deficiencies in existing computational lung models and what we have done in the last couple of years to address those deficiencies and developing the new generation of the whole lung model. If time allows, we will introduce a little bit about the applications like targeted drug delivery, non-invasive diagnosis, and the animal-to-human scale-up. So, talking about the lung diseases, no matter it's like chronic lung diseases like asthma, or it's like lung cancer, or it's infectious disease. So we always want to know that where is the lesion, and we want to deliver the drug directly to those designated lung sites, and do not want to deliver, for example, the chemotherapeutic particles to the healthy tissue to cause side effect. So in that case, we do want to know where are those particles will deposit in localized sites in the lung. By acquiring those information, we do need some high-resolution medical imaging techniques. Unfortunately, till today, the medical imaging resolution is not as high as we want. But if you can see the right-hand side of this slide, when we have a validated computational model, then we can provide high-resolution quantitative results, which definitely can help us to generate more insight and to guide the pulmonary healthcare to be more effective. So usually when we talk about the computational lung model, we always start from CT scan images or MRI scan images. We want to reconstruct our 3D airway as close as possible to the real airway geometry in human body. And sometimes we might want to make it a little bit fancy to add a body shell, but most of the time when we try to simulate inhaled particle dynamics, the body shell is not quite necessary. And the next step is that we want to break this complex geometry into computational meshes. And then it's time for us to start to do the simulation to track the particle trajectories, to track the humidity in the lung airway and some therapeutic gas in the lung airway, as well as the airway deformation, as you may see to the right of these slides. But there are a lot of existing lung models. Nowadays, there are still a couple of deficiencies to limit the physiological realism of those lung models from application. So the first thing is about that lung airway deformation. So most of the existing computational lung assume the airway is just like a rigid pipeline. They neglect the airway deformation kinematics, which just disable our capability to simulate the disease-specific lung condition. The second limitation about the existing lung model is about the whole lung achievement. So our lung starts from the mouth and nose and goes into the alveoli, but when we try to reconstruct the small geometry of those tiny structures, it's really hard to use medical image to do that. Missing those small airways and alveoli will limit us to do the whole lung simulation. So if we do want to know how many drugs or maybe how many virus goes into the deeper airway, we do need a complete route of the pulmonary system. And also, the deposition sites in the lung airway is not the final destination about drugs and about the virus. So it can go into the blood circulation, so the translocation needs to be also simulated. So to address those deficiencies in existing lung model, we try to develop what we call the elastic whole lung model, as you may see here. So it covers from the mouth to trachea, then to bifurcation. After bifurcation goes down to the alveoli, which is also reconstructed by us. The alveoli geometry is to the very right of this slide. There's another example about this simulation. Like this is the simplified whole lung model with the airway deformation. As you may see, the airway is expanding and contracting. Also, the glottis motion has been also included in this simulation. So the airway deformation will be simulated simultaneously with the particle tracking. We also have another elastic lung model, which is 3D, and it's an isotropic lung deformation can be reconstructed. So this is more complex and take more time to finish the simulation, but this is ready to be used too. Talking about the anisotropic airway deformation kinematics. So what we did is just use the pure mathematics to recover this airway deformation from time to time. So what we did is we try to control what we call the nodal displacement on the lung airway geometry from time to time. And this is not only a pure mathematical game, it's also validated with the pulmonary function test data, not only for the healthy lung, but also for the diseased lung, like mild, moderate, and severe COPD conditions. So we do want to make sure that when we include the air deformation kinematics, it's important for us to do that, because when we try to simulate this deformation, it takes longer time for us to finish the simulation. So we compare static airway simulation results and the elastic airway simulation results. As you may see, the particle deposition pattern is quite different, which indicates that the airway deformation kinematics has a significant effect on the prediction accuracy using the computational lung model. So it's definitely necessary for us to include those airway deformation in the simulation. The other thing, like we discussed a little bit, the deposition of those particles in the human lung airway is not the final destination. It can go into the blood circulation and goes to different organs. So we do want to extend our simulation after the particle deposition and make it go to the PK side. We can predict the peak concentration and the average concentration of those drugs or maybe other chemicals in different organs. So those can give us good indication about whether our treatment may cause some overdose potential problems, or whether our dose is not high enough to give an effective therapeutic effect. So the application of the computational lung model, the first one we want to talk about is how we achieve the targeted pulmonary drug delivery. So usually when we take the drugs into the lung airway, like what we showed here, it's just spread everywhere, like 5 lobes without any preference. But sometimes we may want to just deliver the drug to a specific designated lung site. So what we try is something like this. So we think about if we can control the injection at the mouth front to a specific region, can we limit the particle trajectories and the delivery to a specific lung lobe? Like what you may see here, like we release the particles from the yellow region in front of the mouth, and most of the particle ends up in the yellow lobe, which is our left upper lobe in the human body. So we also try something else. Like let's say that we change the release position to the green region at the mouth front, and most of the particles, as you may see from the animation, goes into the right lower lobe. So this gives us the hint that we can achieve the lobe-specific drug delivery by control the particle airflow dynamics in the pulmonary route. And we also think about inter-subject variability and generate different particle release map at the mouth front. As you may see, some of the geometry can give you a nice release map with a large region you can put in the nozzle, but some of them, like the very right one, is just like a really chaos pattern, so it's really hard for us to put the nozzle into a specific region and achieve the lobe-specific delivery. And the future work about this, we always want to think about the lung tumor. So when we deliver the chemotherapeutic particle through the pulmonary routes, we do want all of them deposit on the lung tumor, not on the healthy tissue. So if we can do some further research to try this targeted delivery method, we may achieve this very, very concentrated delivery pattern and to enhance the therapeutic effect but limit the side effect. So the second application is about what we did recently. We try to use computational fluid dynamics and the neural network to create a algorithm to achieve the non-invasive diagnosis and to locate obstruction sites in the deeper lung. So this comes from the idea that, from the fact that occupational exposure can cause a lot of lower lung diseases and lower airway obstruction. But those obstructions are really hard to be detected at the early stage, especially using the existing diagnostic method. So what we think is something like this. So when a person has an obstruction in their deeper lung, when they do expiration, it can cause the airflow velocity pattern shifted from what it should be. Then can we use those velocity distribution in the trachea as a marker to identify or to locate the obstruction locations in small airways? We also think about the possible clinical implementation about this idea. Luckily, we found that there is a type of MRI that can measure the velocity distribution in the trachea. So later on, it's come to this. So we want to use the velocity contour in the trachea as the input into our CNN algorithms, and then the output should be the location of the obstructions. So when we try to develop this algorithm, the first step, of course, we prepare the training and testing database. And those databases are all the simulations from the CFD study. And we choose a subject-specific airway geometry with 44 small airway terminals. And we try to simulate the expiratory flow with 60 liters per minute. When we do this, we block one or two of those small airways arbitrarily. So we create 990 cases in this case. And we use 80% of these cases for training and 20% of these cases for testing. And when we have the database ready, we started to train our CNN-based model. We use two different open source code. One is called YOLO v4, another one is called ResNet 50. And what we did, as we mentioned, we just throw in all those training databases which are labeled velocity contours to train the algorithm. And later on, we use the rest of the 20% of the images to test the accuracy of prediction. And the output is definitely about where is the lung obstruction, whether it's in the left or right or in both lungs. Same thing about the ResNet 50. We do the same thing about training and testing. And luckily, what we see from our results is that our average precision for this testing are all above 93.85%. So those two CNN-based models are really good classifiers to detect the locations of those obstructions using such a non-invasive pattern. So the future about this research is definitely we want to provide more detailed information about the obstruction. Now it's only about whether the obstruction is in the left or right or both lungs. We want to provide more details, saying that if we want to throw in an image and then the output should be which bifurcation is this obstruction located at and in which lung lobe. So application C is about interspecies extrapolation. So there's a lot of animal studies, and we do want to use animal studies to project human responses. But animals are sometimes not 100% same as human. As you may see from those anatomical features, so we use rats to do a lot of experiments, but actually the rat respiratory system is quite different from humans. So using the computational lung model, we can do a better scale up between the animal and human because we can simulate the particle depositions in animal and in human and also the PKs in animal and in human. So in that case, we can do this scale up based on the data considering the anatomical differences between different species. So in summary, so we have been working to establish the next generation lung model for many years. And we see the contribution from this in silico tool that can be on the pulmonary healthcare side and also can be on the health risk assessment side. There are challenges and opportunities. We think that if we can keep improving this modeling framework to make the model more physiologically realistic, in the near future, we can build an in silico clinical trial platform to use such a model to help the pulmonary healthcare, to help the treatment to be more effective and enhance the human life quality. So that's all what I want to say today. And also, you can find more information about this modeling framework using those links. And last but not least, we want to thank all our collaborators, thank all our funding agency, and all my students and private students who contributed to this research area. Thank you very much.
Do we have any questions at this time? Seeing none from the audience, in the interest of time, we want to thank you so much for being with us today, Dr. Fung, and hope you can stay for the rest of the day. Thank you, sir.
Hello, my name is Landon Butler. I am a first-year's master's student here at OSU in the Comparative Biomedical Sciences program, working under Dr. Carrie Pope. I'm here today to describe a study in our lab looking at the interactive effects of diisopropyl and atropine on cholinergic signaling, cholinergic signs of toxicity, forced swim behavior, and monoamine signaling in rats. So a little bit of background. Diisopropyl, or DFP, is a prototype organophosphate acetylcholinesterase inhibitor. Organophosphates are a class of chemicals commonly used as pesticides, plasticizers, and flame retardants. Unfortunately, they are even used as chemical warfare agents. Most recently, the nerve agent sarin was used in the 2017 Syrian attack. Organophosphate toxicity is commonly seen in agricultural settings where risk of pesticide exposure is high. Acute organophosphate toxicity signs typically include convulsions, excessive secretions, arrhythmia, respiratory depression, and more. Acute intoxication with DFP elicits both acute functional and persistent behavioral changes. The standard treatment for organophosphate toxicity is typically the antidote atropine, which is a cholinergic receptor antagonist that reduces acute signs of cholinergic receptor overstimulation. In this study, we evaluated the effects of a single dose of atropine on both acute and delayed effects of DFP intoxication in rats. As our lab has previously reported, persistent DFP-elicited changes in the forced swim behavior test can serve as an effective indicator of depressive-like behavior. Additionally, in this study, we observed the potential changes in monoamine signaling in the frontal cortex. If you'll take a look at this figure here, this shows a cholinergic terminal, with the presynaptic cholinergic neuron at the top and the postsynaptic cell at the bottom. When the presynaptic neuron is depolarized, an influx of extracellular calcium leads to the release of acetylcholine into the synapse. Acetylcholine into the synapse activates cholinergic muscarinic or nicotinic receptors on the postsynaptic cell to modulate its function. Following this, the extremely rapid hydrolysis of acetylcholine by acetylcholinesterase, the degradation enzyme, terminates the signaling. Once hydrolyzed into choline and acetate, the choline is transported back into the presynaptic terminal so that acetylcholine synthesis can reoccur. However, when an organophosphate such as DFP is introduced to the synaptic region, it binds to acetylcholinesterase to block its activity, thus leading to an overaccumulation of synaptic acetylcholine levels that results in the overstimulation of cholinergic signs and toxicity. The methods used for this study included the use of young male adult SD rats were used in the 3 treatment groups, which included the control, the DFP only group, and the DFP plus atropine group. The following evaluations were then conducted: the functional signs of toxicity, which focuses on involuntary movements or convulsions, immobility in the forced swim test at 1 and 4 months following DFP exposure, serotonin, dopamine, and their corresponding metabolite levels in the frontal cortex at hour 4 and 4 months following DFP exposure. This brings us to our first set of results, which measures acute toxicity or involuntary movements of the 3 test groups over time. If you look at the figure shown, the blue spheres represent the control group, and the black spheres represent the DFP-only group. And the red triangles represent the DFP plus atropine group. You'll notice that by hour 1, you'll notice that at hour 1, DFP intoxication, the involuntary movements of the group that received the antidote, atropine, were reduced to that of the control group. However, by hour 4, involuntary movements of the treatment group were almost that of the group that did not receive the antidote at all. Therefore, this shows that atropine only transiently reduces involuntary movements. Atropine has also showed to have little effect on later behavior changes indicated by the forced swim test. The forced swim test is commonly used to evaluate depressive-like behavior. This test includes placing a rodent in an inescapable vessel of water in which they will be recorded and allowed to freely swim or not swim. Later on, these recordings are evaluated to score the test subject's immobility over time. If you look at figure A and B, with figure A representing 1 month post-DFP exposure and figure B representing 4 months post-DFP exposure, you will notice that DFP in both instances demonstrates a higher percentage of immobility. Therefore, atropine did not have a considerable influence on depressive-like behavior at 1 month or 4 months following DFP exposure. When looking at dopamine signaling, you'll notice that at 4 hours post-DFP intoxication, dopamine levels at the frontal cortex were evaluated. Looking at the figure A, you'll notice that this is a measure of dopamine directly, with the DFP group and the DFP plus atropine group having no statistically significant effect on dopamine levels. Figure B and figure C demonstrate the dopamine metabolites, DOPAC and homovinylic acid, respectively. This measure of metabolites indicates the dopamine turnover rate. Therefore, a higher concentration of these metabolites indicates a higher rate of turnover of dopamine. The results shown in figure B and C demonstrate that these 2 metabolites were increased early on by DFP exposure and were not significantly influenced by the atropine treatment. Figure D is actually a function of the two metabolites over dopamine, or rather a ratio of dopamine to the respective metabolites. This suggests that there's an early increase of dopamine turnover in the DFP-only group and not the combination group. Serotonin, or 5-HT, at 4 hours post-DFP exposure is indicated by figure A in the upper left-hand corner. You will notice that serotonin levels are relatively unaffected as compared to the control. However, in figure B, which represents the serotonin metabolite 5-hydroxyindole acetic acid, is apparent that in the DFP and DFP plus atropine group that these metabolites are twice that of the control. The function of HIA to serotonin is shown by figure C. This suggests that early on, with or without atropine treatment, serotonin levels will be modulated by DFP intoxication. Dopamine signaling at 4 months is indicated by figure A in the upper left-hand corner. Dopamine levels were not significantly affected by DFP alone. However, there is a notable decrease in dopamine in the DFP plus atropine group. The DOPAC metabolite was not significantly affected by DFP alone, but had a markedly increase by DFP plus atropine. Homovinylic acid showed no sign of change across the board. Figure D suggests that DFP alone had a slight effect. However, dopamine turnover was largely increased by the DFP plus atropine group. When looking at serotonin levels 4 months post-DFP exposure, you'll notice that serotonin levels were not statistically affected. However, DFP and atropine did show a reduction in serotonin. Once again, HIA levels were unaffected by DFP alone, but increased by DFP and atropine. Graph C shows a clear representation of a persistent increase in serotonin turnover following the exposure to DFP and atropine. In conclusion, a single dose of atropine showed to transiently reduce cholinergic overstimulation signs following DFP exposure. DFP intoxication led to a persistent increase of immobility over the 4-month span with no significant effect from the antidote atropine. While DFP alone did increase dopamine turnover early on, it did not later. Meanwhile, DFP plus atropine had a markedly increase against dopamine degradation later on. The increased monoamine turnover does not appear to be correlated with the persistent behavioral alterations. However, long-term neurochemical effects may be elicited by a single dose of atropine following an acute organophosphate exposure. These studies were generously funded by the National Institute of Neurological Disorders and Stroke via an R21 grant. I'd like to thank the OSU Board of Regents and the fellowship support from the OSU Interdisciplinary Toxicology Program. Thank you. I think that could be an interesting thing to look at. So the typical treatment for an OP or DFP intoxication is typically a single dose of atropine almost immediately, if as soon as possible to the exposure. But that would be an interesting thing to look at. I would like to look at that more.
Do we have any additional questions from our audience? Seeing none, we would like to thank Landon very much for presenting today. And at this time, we will take a short break. I would also remind our attendees, both in person as well as virtually, that each of the abstracts in the compendium do list an email in the event you would like to contact the presenter to discuss additional research in the future. We will see you back shortly, and we will start back up right at 9 o'clock.
Thank you.
We're going to go ahead and get started from our break. Our next presenter is Dr. Randall L. Davis, Associate Dean for the Oklahoma State University Center for Health Sciences. Dr. Davis will be presenting on targeting neuroinflammation with novel anti-inflammatory agents.
Thank you very much for the introduction, and thank you all for being here this morning. The other title for this is The Davis Lab, 15 Years Crammed into 11 Minutes. So hold on, here we go. Everything we care about in the Davis Lab revolves around neuroinflammation. The reason we care about neuroinflammation is that neurological diseases, an array of those, contain neuroinflammation, everything from HIV dementia to neurodegenerative diseases to what I'll be talking a little bit more about today, which is infection, mood disorders, and at the very end, a little bit about neuropathic pain. When we think about inflammation in the brain, that can take a lot of different forms. One of the areas that we're most interested in is the glia, and that is the astrocytes in particular, and I'll talk a little bit about microglia. But basically, these are the cells that obviously have a normal physiological function I've listed here, but they also are first to the scene to deal with whatever insult or damage or infection comes in. So the first part of this talk is really about some of the earlier work that we're still going on today, but characterizing and explaining how a novel anti-inflammatory agent called beta-phenyltretsamine, or beta-FNA, how it can work as an anti-inflammatory agent. I won't take you through all the history of how we got here, but suffice it to say, we kind of stumbled on it, and that is we found here is that this is a drug that it's characterized as an antagonist at the mu opiate receptor. But the data that I'm going to show you here is not using it as an antagonist, it's working in the absence of any agonist. Okay, so there's no morphine, no fentanyl present, just the beta-FNA. And our goal here, of course, is to come up with alternate therapies to treat these disorders. Currently, the treatment out there for mood disorders, anxiety, depression doesn't typically include an anti-inflammatory agent. The thought is if we included one, it might be effective. Our basic in vitro model shown here, we have different cell lines, astrocytes, microglial cell lines, that we can mimic an inflammatory response by exposing those to cytokines, HIV proteins, lipopolysaccharide, and so on. Do that in the presence or absence of our drugs, and then measure markers of inflammation at select times. Kind of a standard piece of data that we've gotten over the years with different cell lines is what I'm showing here. And we're measuring IP10 or CXCL10, which is an inflammatory chemokine, as an indicator of inflammatory signaling in these cells. And basically what we show here is beta-FNA dose-dependently inhibiting the expression of this chemokine. And we induce the expression, in this case, with interleukin-1 beta. We've done this with other cytokines, get the same effect. We've done it with multiple cell lines, we get the same effect. What I've summarized here in the following pop-ups are the take-home message from several experiments that helped us begin to understand the mechanism. First of which I've already mentioned is that it doesn't matter what inflammatory cytokine we've tested so far, we get the same effect. If we wash out the beta-FNA and then stimulate, we still get inhibition. So this is suggesting an irreversible event. If the interleukin-1 beta, or the stimulus, is washed out after a brief period, we've activated the cells, we wash it away, then we add the beta-FNA, we still get inhibition. This, we believe, is suggesting that the beta-FNA is acting downstream of the IL-1 receptor. The most obvious target would be the mu opiate receptor. When we've done the reality is these cells that we've tested in our hands have relatively low expression of the mu opiate receptor. And in fact, when we overload the system with another antagonist, in this case, naltrexone, in excess, it does not change the effect of beta-FNA. Again, all coming together to suggest that the anti-inflammatory effects that we're seeing in these cells is not related to any of the actions at the mu opiate receptor. So what might the beta-FNA be targeting? Our current hypothesis is that it's going at something central to all these inflammatory pathways, which is NF-kappa B. And in fact, in this Western blot here, we've been able to demonstrate that indeed beta-FNA will inhibit the expression of NF-kappa B. And we've done some other tests of NF-kappa B activation, and they all show the same thing. We've done this with P38 and various other inflammatory factors that you see in this diagram. Again, as we go upstream towards the cell receptor, we're getting inhibition by the beta-FNA. So, just briefly to finish up the in vitro part here, this is one of those other inflammatory signals, A20, which is a cytoplasmic ubiquitin modifying enzyme. It's involved in control of NF-kappa B signaling as well as P38, and it also is inhibited by the beta-FNA, which is interesting. We don't know exactly what it means yet, but it gives us more direction. So, we kind of took a few steps back, looking at the basics. What do we know about beta-FNA? Well, we know about it from the mu-opiate receptor literature that its mechanism of action is that it alkylates lysine residues in the mu-opiate receptor. So maybe what it's doing in this system is it could be that it's alkylating other molecules in the signaling pathway that may explain its anti-inflammatory activity. And indeed, we have ongoing experiments to test this. Again, I'm not going through those today, but just showing you a little bit of the targets. TAC1 is one of which we're going to go after and looking at a little bit, trying to identify where the beta-FNA might be acting. So what does this do in vivo? Briefly, in vivo, if we take mice and we inject them with LPS, depending on the dose and the time we look at them, we're going to we'll get sickness behavior early on, and then later more of a depressive-like, anxiety-like behavior. The gist of it here is, if we IP inject these mice with LPS, they get sick. If you inject them right after that, or right at the same time, essentially, with beta-FNA, all this is IP. What we see here is an open field test. Your saline there in green, the mice are moving around freely. In the open field test, LPS, they're sick, they run to the corner and hang out for 10 minutes. And if you give them beta-FNA, they're somewhere in the middle. If we then go in and look at the brain tissue in these mice, what we see is that these inflammatory cytokines, IP10 as well as others, are in fact inhibited in the brain, and it correlates with the sickness behavior. We have ongoing experiments that are addressing this. One of the students in my lab, Stephanie Myers, will be talking about that today in her poster. A little bit about future directions. Again, there's more to do in the cells, there's more to do in vivo, and again, one of the things that's in the poster today is about the timing of administration of beta-FNA and how that impacts it. In the last remaining minutes of this talk, I wanted to go through what we've been working on over the last couple years with a collaborator of mine, Dr. Hugo Arias, and then the bulk of the work's been done by the rockstar postdoc in the lab, which is Dr. Ilyas Marmouzi, and he's here today, and he has a poster that'll go into this more, but let me take you through that briefly. So switching gears a little bit here, we're still in the inflammation realm, but what we're talking about now is a model of chemotherapy-induced neuropathic pain. This neuropathic pain in a clinical setting is often rate-limiting in treatment of things such as colorectal cancer. The drug used is oxaliplatin. Well, what we want to know is where can we block that pain that's associated with the treatment, and can we do it with a safer drug than what's out there right now? The opiates are out there, they can knock down the pain, but we need to stay away from those if we can. So what are some non-opiate options? And that's a little bit about what I'll talk about today. This is just showing a little bit about acute toxicity of this chemotherapeutic agent, but what we're really interested in is more of the chronic setting. And you notice here in the list of mechanisms involved, glial activation is involved. So, with that, the two drugs that we're testing, again, our collaborators have synthesized these drugs. Tongue twister here, but positive allosteric modulation of alpha-7 nicotinic acetylcholine receptors. That's the last time I'll say that because it's really hard. But what we have here is we have a modulating agent that essentially modulates what nicotine or other agonists would do at that site and can potentiate that. Well, what do they do in this setting? Briefly, the model here is take the mouse and inject them over a 14-day period. Then we can test here on the right. We can do a cold sensitivity test, looking at pain. We can do elevated plus maze to look at anxiety behavior, and then the depressive-like behavior with the forced swim test. The drug of interest here is the PAM4, and what we see here is looking at latency in seconds, an indicator of pain. Basically, the oxaliplatin induces the pain behavior, and it is blocked by the PAM4. If we then look at anxiety-like behavior, going through the bars here, we have, this is the open field test, open arms, I'm sorry, back up, open arms in the elevated plus maze. So the time spent in the open arms is what we would expect in a healthy animal, but the one that's treated with the oxaliplatin is going to show this reduced movement or time out in the open arms indicative of anxiety-like behavior, and treatment with the PAM can bring it back to normal. Then lastly, the forced swim test, looking at depressive-like behavior. Again, what we have here is measuring immobility time, is an indicator of depressive-like behavior, and we also see protection from this depressive-like behavior that was induced by the oxaliplatin, corrected by the drug. If we look at the brain, looking at a host of cytokines and chemokines, the theme here as we go across is IL-1 is increased, IL-6 is increased, and CCL2 are all increased. But the oxaliplatin induction is only prevented by the PAM in the case of the IL-1 beta and the IL-6. Conversely, when we look at these same factors in the spinal cord, we see across the board oxaliplatin is inducing inflammation in these, and in fact the PAM4 is protective in all of those. So it seems to be some regional sensitivity to the drug where it might be working. And then lastly, bringing this back in vitro with a human microglial cell line. Very busy here, lots going on. Way to go, Ilias. Has all the controls you would ever want in there. But if we move over to the right-hand side, what we care about is the fact that interleukin is inducing IP-10. The oxaliplatin on top of it at the far right further enhances that expression of IP-10. We can get some inhibition with the nicotine and with the PAM2 in this case on its own. Together, they provide more protection. And then using MLA, an antagonist at the receptor, to confirm that the actions that we have here are indeed through that receptor that I said I wouldn't mention again. And then, if we dig a little farther, fresh off the press here is going into NF-kappa B again, and what we see here is that the interleukin plus the oxaliplatin inducing the NF-kappa B, and we, at the 8-hour time point, this induction is reduced by the PAM. And I believe, in terms of future directions, where we're going to go is we need to get the mechanism of action of these factors. We have got a lot of different ideas planned. I won't go into those, but I'd be happy to discuss later during coffee or other times. And then I hope I'm still in my 11 minutes when I thank the group. This is a look from our lab out across the river, and this is a list of the students over the years that have been involved in helping out, and collaborators, as well as the funding sources. So that is it. Thank you so much, and I'll take any questions you have. How'd I do? Thank you. Thank you for that question. What we have done is we've looked at some of the phosphorylated, the actual activation phosphorylation But I'll be honest with you, we haven't tapped into any larger screening tools for that, but I would love to hear your ideas afterwards on a reliable kit or assay that you feel would do that, because I think that's right where we're wanting to go. Thank you. Thank you. Yes.
This is something that is especially relevant when it comes to human. To follow that, since the veterinary has been there for a while, I did not for a long time. Why is it not become regulated? Is there something I don't know? Because we saw that for the fetal studies, we're not sure how much work we will use for the mouse, but it's also the cells in the mouse.
The dose in the mouse? Yeah, the dose in the mouse it ranges from, if we're doing acute, around 10, is it micrograms per kilogram, up to 50 at the high end. And we do not see a lot of change in that range. So we've also done chronically, or we're working, I have another student in the lab that's going to be doing some chronic studies where we can go lower, theoretically, in the dose, and using an implanted osmotic mini pump to deliver the drug. And again, whether we deliver the drug before the LPS or after are all things that we're looking at right now. Thank you. Thank you all very much.
Thank you, Dr. Davis. Our next presenter is Dr. Matthew Cabine, Assistant Professor with OSU College of Arts and Sciences, presenting on recombinase deficiency in Pseudomonas aeruginosa sensitized cells to antibiotics and stimulates antibacterial killing. Dr. Cabine.
Thank you very much for that introduction, and thank you very much for being here. Thanks to the organizers for inviting me to speak and for organizing this great symposium. Today I'm going to tell you a story from our lab about how we can maybe take advantage of bacterial cells to sort of kill themselves and take others down with them as they go. And I want to start by telling you that most of the work that I'm presenting today was the work of two really talented undergraduates in lab, Adam and Nina, who played soccer together as kindergartners in Richardson, Texas, and came to OSU and joined my lab the first week of their freshman year, barely knowing each other, and who left being great friends and also great scientists. And Adam's getting his MD, PhD at Loyola, and Nina, her PhD at UT Southwestern. So I couldn't be prouder of them. We studied Pseudomonas aeruginosa, which you may know is a notorious opportunistic human pathogen, especially in hospital settings. But what you may not know is that it's also a fierce competitor in the bacterial world. Before humans ever came on the scene, Pseudomonas was fighting with other bacteria through a variety of mechanisms. One is through the secretion of small molecules like redox active antibiotics like the phenazines, which are also virulence factors in humans. Another is the type 6 secretion system, where if another bacterium gets too close to a Pseudomonas cell and provokes it, it'll sort of stick a little dagger into that cell right next to it and inject a toxin into a cell that's adjacent. I don't have time to tell you about it, but it's amazing. You should look it up if I just piqued your curiosity. And another thing that Pseudomonas cells do is produce these things called piocins, which graced the cover of Nature last April. And I'll tell you more about them in a minute. We didn't intend to study piocins. In fact, I'd never even heard of piocins, but we were studying another protein in Pseudomonas, one that hasn't even been named yet, still has a number, 69700. And we were studying a totally different phenomenon, something called biofilm formation, where cells team up to form these communities that are resistant to treatment. And we were trying to figure out what this protein, 69700, did in cells. And so what we ended up doing is transcriptomics to see what changes in RNA levels were caused by deletion of this gene. And we were expecting to see maybe a few genes that would tell us what this protein was doing. And instead, we saw that about 1/6 of the genome was differentially regulated, over 1,000 genes upregulated or downregulated. But what stuck out to us was all these orange ones as being extremely highly upregulated, and we noticed that they were all part of the same gene cluster, and that's the gene cluster that encodes the RF piocins. And so, what are piocins? I had to learn. How do they work? Right, they're like, you can think of it as a bacteriophage tail without a head. So it has this structure that I'll show you a picture of it in a minute. The best studied type are called R-type piocins, and they work as a little contractile apparatus. They attach to a target cell, and they literally stick an iron atom-tipped spear through the cell envelope of the target cell and depolarize the membrane, killing the cell that way. And they are very specific. They are, in fact, species-specific. Pseudomonas uses this mechanism to compete with other cells in the same species, different strains of Pseudomonas aeruginosa. Because they are like phage tails, they're big. They don't get secreted. Their way out of the cell is for the producer cell to lyse, just like phages get out of cells. So the cells that produce these, they bear some cost because they've got to lyse in order for them to get out. And they're typically known to be inducible by DNA damage. So DNA damage induces this response called the SOS response in bacteria. And one of the outcomes of the SOS response, one of the things under the control of the SOS response, among other things, is piocin production. And it was noticed early on that piocins play some role in sensitivity to fluoroquinolone antibiotics, like ciprofloxacin, for example. It's thought that ciprofloxacin, because it causes DNA damage, ends up inducing these piocins to be produced. And one way that ciprofloxacin-treated cells die is because they are lysing themselves to produce these piocins. And here's a picture of piocins here, or a little diagram. Looking like a phage tail, they have these tail fibers that specifically interact with the cell surface of a target cell. They land here like a little lunar lander, and then you can see they contract and just stick a spike through the target cell. And here's a picture of that gene cluster that was upregulated in our studies. And you can see these arrows here indicating the location of the holin and the lysin, the genes that encode proteins that end up lysing the cells that produce these piocins. So we saw that our strain was producing way more piocins than we were expecting at the transcriptional level. We wanted to know, are they actually making the piocins? Can those piocins go and kill target cells? And the answer is yes. We can spread sensitive cells on an agar plate and then take culture supernatants from producer strains and just spot them on top of those sensitive cells, and if there are piocins in there, they will prevent them from growing. So you can see where PA14, which is our wild type, it produces a small number of piocins, so you have a little bit of clearing there in the spot. Our mutant strain here, you can see it's producing plenty of piocins, killing that sensitive indicator strain. Now before I go on, I have to tell you that this unstudied protein was not actually where all the action is coming from. We noticed in our transcriptomic data that this gene is in the middle of an operon, and the genes that are downstream of it, despite the fact that we made a clean in-frame deletion of this gene, are downregulated. And one of those genes here, ZIRC, is a tyrosine recombinase, which is involved in taking DNA apart and putting it back together. So we thought maybe it was this gene that was actually responsible for our phenotype. And the answer is, yes, it is this gene that's responsible for this phenotype. If we take that piocin gene cluster and we put it under a reporter so that we can see whether that cluster is being transcribed or not, you can see that a deletion of ZIRC, that next gene, actually shows much higher expression even than that 69700 mutant we had originally isolated. And you can see that a ZIRC mutant produces plenty of piocins in our sensitivity assay, even more piocins than that original mutant we had studied. And importantly, we can complement both mutations with a second copy of ZIRC, indicating that it really is ZIRC that's doing the heavy lifting for this phenotype. So the hyperproduction of these piocin molecules is because these cells are lacking ZIRC. Now, Nina came up to me and she said, well, because these cells are overexpressing piocins, is it possible that maybe they're also sensitive, hypersensitive to fluoroquinolone antibiotics? And I said, probably not, but let's do the experiment. But she was right and I was wrong, that if we give cells a sub-inhibitory concentration of ciprofloxacin, usually it slows down their growth just a little bit, so those are the open circles here. But if we take our ZIRC strain and we give them ciprofloxacin, then they grow much more poorly, and if we complement back that gene, they return to pretty much wild-type-like growth. So not only is this deletion in ZIRC causing hyperproduction of piocins, it's also causing hypersensitivity to ciprofloxacin, and more than that, when we produce or when we treat these Xerces cells with ciprofloxacin, then they make even more piocins. So we have all these different phenotypes, and we wanted to know, is the pathway by which these piocins are produced the same as the canonical pathway, or is it something new? And so canonically, I told you it's piocin production is typically induced by DNA damage. And what happens is that there's a repressor protein called part R, which represses the transcription of an activator protein called part N. And in the presence of DNA damage, another protein called REC-A becomes activated and activates cleavage activity of part R. Part R gets cleaved, no longer repressing part N. Part N can be made, and then part N goes and binds to the promoter of the piocin gene cluster to activate that transcription. So we wanted to know, this canonical pathway, it depends critically on REC-A and on part N. And we wanted to know if in our alternative pathway, does it also depend on these two proteins? And the answer is REC-A is totally dispensable in Xerces deletion strains. We see the exact same amount of piocins being expressed and produced if we delete if we delete REC-A. But if we delete part N, we see production and expression of piocins drop almost to zero. So what we have here is an alternative pathway for piocin expression that does not depend on DNA damage, does not depend on REC-A, yet still does have in common with that canonical pathway the requirement for that activator part N. Now at this point we wanted to do something that not every Pseudomonas biologist does, and that is look at cells in the microscope. We wanted to know, because this phenotype is potentially deleterious, because producing piocins imposes a cost on cells, makes them lice, could every cell be doing this? Probably not. So we put in a reporter for piocin expression and then looked at cells at the single cell level. And so you can see in wild-type cells here, almost no cells are producing piocins. Very rarely do we see a dimly fluorescent cell, and fluorescence corresponds with piocin production. Whereas in our Xerces cells, we see a bimodal distribution where most cells are off for piocin production and some cells are bright, indicating that they are producing piocins, and we see that happens over a wide range of fluorescence values. So we wondered what was happening with these ones of intermediate fluorescence. We thought maybe they were on the path to fluorescing very strongly and then maybe ultimately lysing. And so to answer that question, we wanted to just look at a time lapse here. So I'm going to show you a quick video where you see that this cell turns on brighter and brighter, and then pop, it explodes. I'll show you that one more time. This is in minutes here, 10 minutes intervals. Bang, the cell explodes. And so what's really happening is that in a population of cells, there's some subset of cells that is turning on piocin production, and then it ramps up. It gets brighter and brighter and brighter until the cells ultimately lyse. And you can see that in this time-lapse series here, where these cells, something happens early on in these cells before we can even detect GFP. All these cells turn on more and more brightly for piocin production until pop, they lyse. Now there's much more to this story, I just don't have time to tell you, so I'm going to summarize what I've talked about so far. That in Xerces cells, we see this strong upregulation of these piocin complexes, and this non-canonical upregulation we see is independent of REC-A, so independent of DNA damage, but it does depend on part N. Production of these piocins is heterogeneous across the cell population. A small subset of cells are really strongly expressing piocins, and those Xerces cells are most sensitive to ciprofloxacin, and when we treat them with cipro, they also produce more piocins. And so our ultimate goal and excitement with this project is that we may be able to take advantage of this phenotype, because we have two effects: a primary effect where we sensitize cells to ciprofloxacin and also make them kill themselves through the production of piocins, and then a potential secondary effect where as they die, they're releasing piocins that can then go kill other Pseudomonas strains that are in the vicinity. And so we think of it sort of like an Old West shootout, where all the bad guys, all the Pseudomonas cells will fire at one another with these piocins, and the good guys can just lay low, and then when the smoke clears, the bad guys are dead and the good guys are still there. This is a picture of our lab before the pandemic started. I'm grateful to support from the National Institutes of Health and from my department, and I'd be happy to take any questions you have. Thanks. He's my department head, but he's not a plant.
You know, somebody has to ask you a question, right? So is part N a transcriptional activator?
Yes.
Does it bind and, okay.
Yeah, question is part N a transcriptional activator? And the answer is yes. That was known before, but we think that it's also activating things that have not been known before. I just didn't have time to talk about them today.
What's known about mechanisms of resistance to piocins?
Great question. Question is, what's known about mechanisms of resistance to piocins? They show high similarity to bacteriophages, so they're thought to use core LPS as a receptor. And so the same mechanisms of resistance that would apply to phages would apply to piocins. So if a target cell does not have the appropriate LPS structure, a particular piocin sort of isotype will not attack that cell. Make sense? Yep.
Nice talk again. I have a question about because most of this work you've done by deleting some of these legislative elements, but in normal circumstances, we either in the environment, what have you found, what really triggers the production of piocins?
Say it again, what?
What triggers the overproduction of piocins without the addition of these regulatory elements? For example, you've done addition of part 1, SACs, those are regulatory elements, and even 67900. But under normal circumstances, what will trigger the production of piocins?
So the question is, what would typically trigger the production of piocins?
Yeah, without deleting these.
Without having any of these deletions. Yeah, it would be DNA damage. It's the only, right now, the only known pathway until we uncovered this alternative pathway is DNA damage. I didn't have time to tell you today, but that was one of the first things we looked for. We thought maybe we're just when we delete this gene, we're causing DNA damage. So we looked for all the genes that would be the usual suspects that would be upregulated in DNA damage, and none of them are upregulated in our strain. So we think we're looking at a totally separate phenomenon. Thanks very much.
Thank you, Dr. Kabin. We will take a short break now and come back just after 9:45, and we will begin session 2 at that time.
Oh, that's the end of the session.
Yeah, department head followed by Nobel Laureate question. Well done. I'm particularly interested in it. I'm glad they asked questions because mine. Hi. How do you ask to go to roughly the next door and then come back? There's kind of a wood appearing door. Push it. Don't turn the handle. Oh, I turned the handle. First presentation, of course, is the Humanimal Trust, and I'll just plan on starting it from here. We did test it this morning, and it worked fine. So if you want to stay at the table and announce, and I will start it, and then we'll go on once we're done with that. Does that sound okay?
Yeah, that's work.
These microphones are, they allowed us because we will be back here, maybe this is on, and this is off. Okay. So the pointer is on. I'm going to turn the pointer off for the first presentation, but it is here, and then I found that it's been moved. I'm going to try to keep it, if they're not going to use it, I'm going to try to start it down here in the corner, if that makes sense. Otherwise, it ends up as a red dot in the middle of their slides. I think we're okay.
All right.
You good?
Yeah.
Okay. I'll introduce you.
Okay.
Just briefly.
Okay.
And then I'll turn it over to you.
Sounds
Is that
good.
okay?
And you want me to stay over there, right?
Yeah, I think so for the moment. We might do that kind of together, side by side, if that's all right with you. Yeah. And then I'll introduce you. That way they can see you, because on the live stream they can't really see us back here.
Okay. Gotcha.
And so that way they can see your shining face. So I like the green. The green is very pretty. I have to go to the restroom. Kind of getting into a bit of a group. There were a few bumps of just not knowing. So, all right. studied at, he went to Royal Veterinary College, so yeah. And then, actually, he's a double doctor, because he has a PhD, too, so. One of them. Are you a double doctor, too? No, no. Oh, okay. I'm just PhD. Should start right back at 11. Yep. Okay.
Dr. Roberta?
No, Dr. Roberto's not here, right?
So you want
From
to
the
do
Humanimal Trust? It's a pre-recorded one.
But do we need
Yeah,
to
yeah.
tell those that did it?
No, it's pre-recorded on the presentation.
Oh, so we just, but you're
Yep.
going to.
So are you
Yep.
going to
He is still attending, like he's watching his talk, so you
Okay.
just kind of thank him.
Yeah, if you'll introduce him, I'll stand up there, push play. Okay.
Just like really, and one announcement we can make. We did a MOU with Interact.
Okay.
Recently, and thank the Animal Trust.
Okay.
One health and one.
Got it, got it. Okay, I'm ready if you are. Ari, are you in good shape? Okay. Good morning. We're going to go ahead and get started back. It's my pleasure to serve as co-moderator. I'm Dr. Rosalind Biggs from the College of Veterinary Medicine, and joining me for this second session of the Interact Symposium is Dr. Dolores Vasquez from OSU Center for Health Sciences. We will get started here shortly, and thank you for your attendance.
Welcome everyone to this second session of this vibrant and very interesting Interact Symposium. It's my great pleasure to introduce you to Dr. Roberto Larrañón, and he's going to talk about the human-animal trust.
Everybody, I'm Roberto Larrañón, and I'm the chair of trustees for Humanimal Trust. I'm a veterinary microbiologist based at the University of Surrey in Guildford, UK. And today I'm going to speak to you about One Medicine and the work of the Humanimal Trust. But before I get started, I'd like to thank the organisers for inviting me to this wonderful meeting today. And I look forward to giving you some insight into the work of the Trust. So with no further ado, I'll get started on my first slide. So One Health and One Medicine, what is it? Well, there are two concepts that are interrelated, but are synergistic rather than synonymous with each other. One Health, in its present form, since the early 2000s, was defined by the World Organization for Animal Health, the OIE, as being that human health and animal health are interdependent and bound to the health of the ecosystem in which they exist. And interestingly, the foundations of One Health lie in a much older concept, One Medicine. And One Medicine does not seek to undermine the more well-known One Health concept, but merely goes beyond in its scope. So One Medicine, what is it? Well, with One Medicine, the emphasis is on the close interdependence and similarities between human and veterinary medicine. And that might be general medicine of humans or domestic animals, or indeed free living animals. And if we look back at the work of Calvin Schwab in 1964, we can go right back to anthropology, linking in with human medicine, veterinary medicine, animal husbandry, and the study of free living animals, and bringing those all together under the ologies of general medicine. And that was the foundation of One Medicine. However, the Trust's definition of One Medicine is a little different. And we look at One Medicine where it drives collaboration between vets, doctors, researchers, and the allied health and scientific disciplines, so that all humans and animals benefit from sustainable and equal medical progress, but not at the expense of an animal's life. And if we look back at some of the history, we think very much about Rudolf Virchow's work, where he spoke about between animal and human medicine, there is no dividing line, nor should they be. And that very much links and resonates with the Trust definition. So what brings human and veterinary medicine together? Well, many different things, but most importantly, a common language, but actually also there is high level of patient care across the two disciplines, common values, love, empathy, and compassion, the history behind the medical progress and embryology, comparative anatomy and physiology, the comparative disease states, oncology or infectious disease, and indeed things like zoonoses, where we see infections being spread from humans to animals and animals to humans and vice versa. So zoonotic diseases such as things like MRSA, public health and epidemiology, and technological advances, we mustn't forget those, because this is how the science advances. Whether it's in human or the veterinary side, those advances are really important. And we need to bring them together in order to advance both disciplines at the same rate. So what are the benefits of a one medicine approach? Well, I've touched on some of those already, but to start off with, advances in human and animal medicine can be achieved more quickly with closer collaboration and mutual learning. We believe this will lead to fewer laboratory animal tests until one day they may be obsolete or very rare. We all share responsibility for creating a fairer society with better opportunities to benefit equitably from medical progress for both humans and animals, and progress the medical sciences. And we can already see a lot of this happening in the literature. We see many publications where one medicine is mentioned, particularly when we're looking at diseases such as osteoarthritis that we see in animals and humans, oncology and infectious diseases. And more recently, we've seen a lot around antimicrobial resistance and the interrelationship between animals and humans, particularly where we're looking at zoonotic diseases, which have been spread from animals to humans or humans to animals in reverse zoonoses. So a little background to Human-Animal Trust and its origins. It was founded in 2014 by Professor Noel Fitzpatrick, a vet who experienced a deep divide between human and animal medicine and saw how unfair this was and could be. He was particularly frustrated by the lack of opportunities to share what he was learning day to day in practice, or to benefit from the relevant learning that was happening in human medicine. So he decided to create his own platform, Human-Animal Trust, which has now developed into a very well-known trust, which I hope I can tell you a little bit more about in the coming minutes. One of the main areas within the trust is thinking about the 3 R's. Replacement, where we replace animal studies with other methods such as in vitro studies. Reduction, as many trials as required, as few as possible, to reduce the number of animals that have been used. Refinement, minimizing the severity and the stress of studies that are undertaken on animals. But we also have within the trust a 4th R, reciprocity, where we look at collaboration, interdependency, and empathy linked to the other 3 R's. And we have 5 different flagship areas which we fund and we're particularly focused on within the trust. And these are as follows: infection control and antimicrobial resistance, cancer, brain and spine disease, bone, muscle and joint disease, and regenerative medicine. And all of those link together, and we link those from a veterinary perspective and a human perspective. And we also think about how the environment interacts with these different disease states. Now I'd like to tell you a little bit about the different projects that are ongoing and that have taken place a few years back within the trust. So one of the current ongoing projects is understanding how osteosarcoma spreads. And this is a project at the University of Cambridge with the principal investigator being Professor Matthew Allen. And we know that we see an occurrence both in humans and dogs with a high incident rate, approximately 10 times higher. And if we think about how we want to investigate this project is looking particularly at investigating better ways to predict which patients have a higher risk of their cancer spreading. A second project is in collaboration with Action Medical Research for Children, and we have 2 projects here, in fact. The first one is preventing infections and reducing the risk of antibiotic-resistant infections in critically ill children. And the second one is looking at juvenile idiopathic arthritis and thinking about personalising drug treatment to improve eventual outcomes and well-being. Some of the previous projects that we funded include developing liquid biopsies for canine patients, and this was with Professor Joanne Morris and Dr. Tomoko Iwata at Glasgow University, and they used liquid biopsies to detect fragments of DNA from tumour cells. And a second project was a PhD studentship based at the University of Surrey, And in this project, Dr. Lucy Grist specifically looked at bacteriophages as an alternative to antibiotics, and specifically focused on E. coli, which was causing infections in dogs, and trying to reduce the resistant bugs by using an alternative to antibiotics. So what is the world we want to see in the future? We want to have human and animal health demonstrably improved as a result of one medicine. One medicine be widely understood and embraced, have collaboration between human and animal medicine as the norm, and finally, there be sufficient funding and robust public policy to support collaboration between human and animal medicine. And I'd like to finish off by acknowledging Interact Institute, which we're now formally collaborating with, and also the Animal Trust funded students and researchers who undertook much of the work which I presented today. So thank you very much for listening. I'd be very happy to answer any questions if there are any, and thank you very much.
Our next speaker is Dr. Jun Cheng from the Department of Biochemistry and Molecular Biology at Oklahoma State University.
As we prepare for our next presenter, we also want to recognize Humanimal Trust with a recent memorandum of understanding with Interact and the College of Veterinary Medicine at Oklahoma State, and we are excited for that new partnership.
Stand fairly close to the podium. There's your mic. Okay, thanks. Okay.
Good morning. At the beginning, I want to thank the Symposium Organization Committee for giving me this opportunity so I can present my research here. Today, my presentation will talk about therapeutic application of extracellular vesicles in tuberculosis. Tuberculosis airborne disease is caused by mycobacterium tuberculosis. Mycobacterium tuberculosis, we also call it MTB, is an intracellular bacterial pathogen. It mainly infects alveolar macrophage in the lung and finally causes lung disease. When MTB enters the lung, it can be uptaken by different immune cells, including macrophages and dendritic cells. And then they can form a structure in the lung called granuloma. Finally, MTB can cause latent TB or active TB, two different types of infections in a human. According to WHO reports, there are about 10 million people that have active TB each year. It causes around 1.5 million deaths each year. When MTB enters the lung, as I mentioned, it can be uptaken by macrophages, alveolar macrophages. It can survive in alveolar macrophages in a structure called phagosome, or it can be degraded in a structure called phagolysomes by the fusion between phagosome and lysosome. Or MTB can activate autophagy and then degrade in autophagosome. Extracellular vesicles are vesicles released by eukaryotic cells. There are two main types of extracellular vesicles, macrovesicles or exosomes. Our study focuses on exosomes. Exosomes are nanoscale mammary vesicles. It formed in called multivesicle bodies within eukaryotic cells, including mammalian cells. And then these vesicles are released into the extracellular environment by the fusion between MVB and the cytoplasmic membrane. Our study indicates MTB-infected macrophages can release exons into the extracellular environment, including the blood and serum. These exons include MTB components, including MTB protein and lipids. So right now, come to our topic. Our previous study also found MTB can release RNA into the cytosol of the macrophages. It indicates this RNA can activate the cellular response in host cells. So we have a question here. If this MTB-released RNA can be packed into the exons node and further activate neighboring cells or receiving cells. To test our hypothesis, what we do is we look at MTB RNA in the exons we isolate from MTB-infected macrophages by qRT-PCR. Based on this result here, we can see our MTB RNA in the exons we isolate from MTB-infected macrophages. However, not in the exons we isolate from uninfected macrophages. As we know, the viral RNA can activate interferon beta production or type 1 interferon production in host cells. So we ask if the exons we isolate from MTB-infected macrophages also can activate interferon beta production not in naive macrophages. So here we can see, based on our qRT-PCR result, we can see exons from MTB-infected macrophages can induce interferon beta production in naive macrophages. We also can see this dose-dependent response. When we knock down RIG-1 mouse, these are two very important components in the host cytosolic RNA pathway. We can see decreased interferon beta production in macrophages. They are treated using exons from MTB-infected macrophages when compared to control cells. So it means both RIG-1 and MAVs, these two genes or proteins are important for the exon-induced interferon beta production in macrophages. A similar result was detected when we used MAV-deficient mouse bone marrow macrophages. So here we can see a decreased interferon beta production both by qRT-PCR or ELISA in macrophages treated using exons from MTB-infected macrophages when compared to the wild-type cells. Next, we look at the downstream factors of the MAV-dependent signaling pathway. As we know, in a contact viral infection, TBK1 can be activated by the RIG-1 MAV-dependent RNA pathway. So what we do is we look at the TBK1 activation in macrophages post-exon treatment. TBK1 is a protein kinase can autophosphorate. So we look at the TBK1 phosphorylation by Western blot. So here we can see an increased TBK1 phosphorylation in macrophages treated using exons from MTB-infected macrophages. When we knock down MAVs, we see a decreased TBK1 activation in macrophages. So it means the MAV is required for exon-activated TBK1 phosphorylation or activation in macrophages. Next, we look at another downstream factors of the MAV-dependent signaling pathway, IF3. IF3 is a transcription factor. This can be phosphorylated by TBK1. After phosphorylation, IF3 is translocated into the nucleus and then bound to the promoter of interferon beta and drive interferon beta production. So what we do is we look at the IF3 translocation or abundance in the nucleus in macrophages after exon treatment. Similar to the TBK1, we can see increased IF3 in the nucleus of macrophages after exon treatment. So these exons we isolate from the MTB-infected macrophages. When we knock down MAVs, we see a decreased IF3 abundance in the nucleus. So it means similar to TBK1, MAVs is also required for the IF3 activation in macrophages. To further confirm our result for the IF3, we look at the IF3 nuclear translocation by fluorescence microscope. Here we can see when we use the exons from MTB-infected macrophages, we can see the exons can increase their nuclear translocation of IF3 in naive macrophages. Here we use DAPI as the marker for nucleus, so you can see the increased colocalization between IF3 signal and DAPI in naive macrophages after exon treatment. When we knock down RIG-1 and the MAVs, we see a decreased colocalization of the IF3 and the DAPI. So it means IF3 is not translocated into the nucleus. So altogether, right now, we know the MTB can release RNA into the macrophages, and this RNA can be packed into the exons. These are extracellular vesicles. These vesicles can further uptaken by the leucemia cells or nearby cells and activate RIG1, MAVS, TBK1, and Ir3 dependent type 1 interferon production in macrophages. So next we ask, besides type 1 interferon production, if the exons also play other roles or not in the context of MTB infection. So what we do is we look at the MTB growth or survival in macrophages. So these macrophages were pre-treated using exons from MTB-infected macrophages. However, we didn't see a difference between the exons from MTB-infected macrophages or exons from uninfected macrophages. So it means the exons is not required or couldn't activate anti-MTB response in macrophages. So when we see this result, we think about what is a possible reason. So we hypothesized maybe a second or third signaling pathway is required, so it works together with exons to regulate anti-MTB response in macrophages. Interferon gamma, so this is a hallmark cytokine in an active TB patient. You can see very high interferon gamma level in the patient serum. So what we do is we add the interferon gamma into the cell culture together with the exons from MTB-infected macrophages. So here we can see the combination of the interferon gamma and the exons significantly decrease MTB growth in macrophages. So it means exons work together with, at least with the interferon gamma to inhibit or activate MTB, anti-MTB response in macrophages. Next, we want to know what is the mechanism, how the exons activate anti-TB response. As we know, in every macrophage or in the host cell cells, MTB can survive in the phagosome and degrade in the phagolysomes by the fusion between the phagosome and the lysomes. So we hypothesized maybe the exons can activate the phagolysome maturation. To test our hypothesis, what we do is we look at the LAMP1 colonization with MTB in macrophages in the presence of the exons. LAMP1 is a marker for phagolysomes. So here we can see exons from MTB macrophages increased colonization of the LAMP1 and MTB. So it means MTB is trafficked or delivered into the phagolysomes and degraded. When we use MAV-deficient mouse macrophages, we didn't see this increased phagolysome maturation in macrophages. So it means exon activated anti-MTB response depends MAVs-mediated pathway in macrophages. When we look at the TBK1 activation in macrophages, we see exons from MTB-infected macrophages can activate TBK1 activation in macrophages. TBK1, this is a very important or key component in autophagy, and then further activate the downstream factors or component. So we ask if the exons also activate autophagy. So this is why we can see the increased phagosome lysosome maturation in macrophages. What we do is we look at the LC3 colonization with the MTB. LC3 is a marker for the autophagy. So here we can see the exons from MTB-infected macrophages do activate the LC3 colonization with MTB. So it means exon can indicates exon can activate autophagy. However, when we use the MAV-deficient mouse macrophages, we didn't see this increased colonization between LC3 and MTB. So it means the MAV exons activate LC3 modification depend on MAVs-mediated pathway in macrophages. However, when we look at the ubiquitin colonization with MTB, we didn't see the difference. For the classical autophagy is ubiquitin dependent, so this is why we use ubiquitin to test the autophagy activation by exons. As I mentioned, we didn't see the difference between the untreated cells or exons from MTB-infected macrophages. So it means exons from MTB-infected macrophages couldn't activate ubiquitin-dependent autophagy in macrophages. Recently, it was reported MTB infection can activate LC3-associated phagocytosis in macrophages. And after activation, LC3-associated phagocytosis can activate NADPH oxidase in macrophages. NADPH oxidase include 5 different subunits and 2 membrane subunits and 3 cytosolic subunits. So what we do is we look at the colonization of NADPH oxidase with MTB in macrophages after exon treatment. So here we use the 1 membrane subunit of NADPH oxidase as our report, GP91. Another name of the GP91 is NOX2. So here we can see exons from MTB infected macrophages can increase the colonization of the NOX2 with MTB in macrophages. So it means exons from MTB infected macrophages can activate LC3-associated fagocylation maturation in macrophages. We didn't see this increased NOX2 colonization with MTB in MAF-deficient mouse bone marrow macrophages. So right now we know except activate LUG1, MAF TBK1-IF3 dependent type 1 interferon production in macrophages. Exons from MTB infected macrophages also can activate exons from MTB infected macrophages also can activate LUG1 MAF-dependent LC3-associated fagocylation maturation in macrophages. So next we wanted to know stuck here. Yes, okay. Let's click. Okay, let's go to it right now. Okay. So next we wanted to know if the exon released by MTB infected macrophages can used as the potential anti-TB therapy or not in the future. To test our hypothesis, what we do is we look at the efficacy of the exons together with anti-MTB drug, moxifloxacin. Moxifloxacin is a second-line anti-TB drug. So here we can see when we combined moxifloxacin with the exons, it significantly decreased MTB survival in macrophages when compared to the drug alone or exon alone. We didn't see this increased MTB killing in macrophages in MAF-deficient mouse bone marrow macrophages. So it means this anti-MTB response depends on MAF's mediated anti-TB response. We next look at the efficiency of the exons in mice. In vivo, what we do is we infect the mice using MTB for 3 weeks. After 3 weeks post-infection, we treat mice using moxifloxacin for 2 weeks. After 1 week treatment, we inject 1 dose of exon into the mice. And after 2 weeks treatment using the drug, we look at the pathology of the lung and MTB survival in the lung and the spleen. So here we can see the combination of moxifloxacin and exons. They decrease the lung lesion when compared to the drug alone or exon alone. So it means the exons together with the moxifloxacin can decrease the lung pathology. When we look at the MTB survival or burden in the lung spleen, we can see the combination of the moxifloxacin and the exons. They decreased MTB growth in the lung and the spleen when compared to the drug alone or exons alone. So altogether, we found MTB can release RNA into the cytosol of macrophages, and then this RNA can be packed into the exons. Released exons can activate LUG1 MAF-dependent type 1 interferon production, and meanwhile, it also can activate LUG1 MAF-dependent LC3-associated fagocylation maturation. Finally, I want to thank all the members in the lab and our collaborators, Dr. Jeff Shorey and Dr. Lin Lu. I also want to thank Dr. Jerry Malaya for his support for our project. Finally, I want to thank Oklahoma Center for Respiratory and Infectious Disease and NHP20 grant. Thank you.
So I have two questions. One is more understanding of how these RNAs are being uptaken by the macrophages. So the EVs, which has both protein, lipid, and RNA, when it fuses to membrane, it would fuse with the endosomes. So that also would have RNAs in it. So how come these RNAs are not degraded in the EVs?
So in general, the exons were very similar to the cells. So actually, we don't know the mTBRNA in the exon is a fragment or full length. We don't know.
But the RNAs which are there in the endosomes does not, it would degrade. It has an exosome activity, so it will degrade all the RNA regardless of what size it would be.
Okay, can you repeat it once more?
So when the EVs fuse with the forms a lysosome.
Yes.
Because of the pH, it will activate all the RNAs. And if the RNAs are activated, I don't understand how these MTV's RNA still survives in the exosomes, like when it's inside the cell, so that the RIG-I MAF pathway gets activated.
So actually, right now we are not clear if the RNA is degraded or not in the exosomes.
Okay.
Yeah.
I'll talk about it later. The second part was, so you talked about RIG-I MAF pathway, but there are MDA5 sensors also. Like other than RIG-I, there are other sensors
for RNA.
So it was kind of striking. If you take away RIG-I, you didn't see any interferon, IF3, also the IL-6 expression levels. So I was wondering if you have tested MDA5 also.
Right now, for this study, we focus on F3.
No. So when the RNA is there in the cytoplasm,
yes,
it will be recognized by RIG-I.
Yes.
And MAD5.
Yes.
There are two sensors.
Yes.
Which will then signaling pathway would be for MAF.
Yes.
To activate or phosphorylate IF3, and then the pathways, right, downstream.
Yes, yes.
But there are two sensors.
Yes, it's a very good question. So actually, for this exon study, we didn't look at MD5,
yeah,
but we have another paper. We looked at MD5 by the co-IP experiment. We look at the mTBRNA bound to the RNA sensor. We found the RIG-I can bind to the mTBRNA, but in our study, we didn't see the MD5 bound to our marker, our RNA. But we couldn't exclude the possibility of MD5 bound to the mTBRNA in macrophages.
That's good, yeah.
Okay.
What exciting conversation. I encourage you to keep it at lunch. Thank you so much, Dr. Yun.
Thank you.
Okay, so our next presenter is Dr. Craig Miller. He's an assistant professor and co-director of Oak Ridge Immunopathology and the College of Veterinary Medicine.
Thank you for the introduction. And thank you for Interact for the opportunity to be here. Today I'm going to give you a brief synopsis of the work my lab has done to develop a feline model for SARS-CoV-2 infection. Now, when developing a new animal model that accurately replicates characteristics of human disease, it's important to start with what we know of the disease in human patients. Well, we know that SARS-CoV-2 utilizes ACE2 as a receptor for entry into cells, and we know that ACE2 is highly expressed in immune cells, endothelial cells, and epithelial cells, and a variety of organs, including the lung, kidney, and gastrointestinal tract. We know that the average time from exposure to symptoms is 5 days, and we know that the most common symptoms in hospitalized patients is fever, dry cough, shortness of breath, and fatigue. We also know that the major clinical pathologic phenotypes of hospitalized COVID-19 patients are pneumonia, prothrombotic coagulopathies, and progression to acute respiratory distress syndrome, or ARDS. Now, ARDS is a clinical syndrome of respiratory failure that we characterize by a rapid onset of acute inflammation and fluid buildup in the lungs, and caused in this case by viral-induced vascular injury and damage to the alveolar epithelium. This results in immune activation and deposition of hyaline membranes, which are characteristic features, shown here, of diffuse alveolar damage. Other pathologic hallmarks that we observe in human COVID-19 patients include alveolar histiocytosis, perivascular lymphocytic infiltrates, pulmonary edema, bronchopneumonia with an influx of neutrophils and necrotic cellular debris into the airways, and fibrinoid vascular necrosis and thrombosis. Now, when selecting an appropriate animal model, there are specific guidelines that we should consider to ensure that the model accurately reflects human disease. In this case, we considered the ideal model for COVID-19 was one that mimicked human disease in the route of infection, severity of disease, the expression and distribution of the viral receptor, and then a correlation between viral replication and disease severity. We looked at these criteria and thought, why not the domestic cat? Now, as we were getting started with these studies, previous studies had shown that cats are susceptible to natural SARS-CoV-2 infection via the intranasal or intraoral inoculation routes. Cats became infected, they shed virus, they could transmit the virus to their cat, and they produced antibodies that could then protect from infection. But cats in these studies exhibited minimal to no clinical signs and minimal to mild pulmonary lesions. And importantly, ARDS was not observed with intranasal or intraoral inoculation. So we took a look at these limitations and thought about ways that they might be overcome. So in our study, we utilized 18 domestic cats, and we intratracheally inoculated 12 of them with 1.2 times 10 to the 6 TCID50 of the Washington isolate of SARS-CoV-2. Now, this was about twice the dose as the previous studies. We also inoculated 6 cats with sterile media, which served as uninfected controls. We necropsied a subset of these cats at day 4 and day 8 post-inoculation and collected samples for analysis. Now, in order to fully evaluate lower respiratory disease and systemic clinical signs in a BSL-3 environment, we developed a novel clinical scoring system that we could utilize under the constraints of our PPE. Cats each day were scored by a licensed veterinarian, our very own Dr. Jennifer Rudd, and then assigned a score from 0 to 2 in each one of these categories shown here on the left. These scores were then totaled to provide, summated to provide a total score per cat per day. Compared to sham-inoculated cats, SARS-CoV-2 infected cats exhibited significant degree of clinical signs at day 4, 5, 6, and 8. Clinical signs peaked at day 4 and continued throughout the endpoint of the study. The most prominent clinical signs that we observed were lethargy and increased respiratory effort, as well as pyrexia or fever and coughing, which accurately reflect the most common clinical signs observed in humans, which are fever, dry cough, dyspnea, and fatigue. At post-mortem evaluation, marked pulmonary consolidation, pulmonary hemorrhage, and edema were observed in SARS-CoV-2 infected cats. And histopathology revealed that pathologic lesions in SARS-CoV-2 infected cats closely mimic those observed in hospitalized COVID-19 patients. At day 4, the lungs of SARS-CoV-2 infected cats exhibited multifocal areas of alveolar inflammation and necrosis with fibrin deposition, alveolar histiocytosis, and perivascular lymphocytic infiltrates. The alveoli in these cats' lungs were frequently filled with large amounts of pulmonary edema, shown here in this inset, and there were multifocal areas of hyaline membrane formation, shown here, all corresponding to pathologic lesions observed in hospitalized COVID-19 patients. At day 8, pulmonary edema and perivascular lymphocytic infiltrates were observed in these cats as on day 4, in addition to pulmonary thrombosis and occasional respiratory syncytial cells as observed in human COVID-19 patients. When we quantified viral RNA in feline tissues by droplet digital PCR, we saw that viral loads increased over time in tracheobronchial lymph nodes while simultaneously decreasing in the lung, indicating clearance of virus from the lung over time, which was not surprising. But when we looked at ACE2 expression, feline ACE2 expression in feline tissues, we observed that the ACE2 expression in nasal turbinates was interestingly higher than in the lung. Overall, ACE2 expression was significantly upregulated at day 4 in COVID-2 infected cats when compared to the sham-inoculated cats and cats with SARS-CoV-2 at day 8. We recently published these findings in a special edition of Viruses and are currently working to define key factors of immune dysfunction that contribute to COVID-19 to COVID-19 progression. Our preliminary data shows that pro-inflammatory cytokines are upregulated in cats during acute SARS-CoV-2 infection, and many of these cytokines, such as IL-1B, IL-6, and IL-8, are also upregulated during cytokine storm syndrome in hospitalized COVID-19 patients. Now, during our studies, we noticed that our viral loads in the lungs of our infected cats were several orders of magnitude lower than previous studies, such as this one shown here by Gaudreau et al. Out of Kansas State, even though our dose was approximately 2 times higher than theirs. Now, because ACE2 expression is significantly lower in the lung than in the nasal turbinates, we hypothesized that our lower viral loads in our model corresponded to bypassing of the upper respiratory tract via intratracheal inoculation, and that intranasal inoculation would result in higher levels of viral replication. So we conducted a follow-up study to evaluate the role of the inoculation route on disease progression and viral infection kinetics. In this study, we utilized the Delta variant of SARS-CoV-2, and we just completed this study last month, in which we inoculated 12 cats via both intratracheal and intranasal inoculation. We also inoculated 12 cats with intratracheal inoculation alone, and 6 cats with sterile media, which served as uninfected controls. This time we took the study out to 12 days post-inoculation, and it's important to note here that the dose we used in this study was approximately 10-fold less than we had used in the previous study. However, cats exhibited similar clinical signs, and the clinical signs were significantly elevated and peaked around day 4 and day 5 post-inoculation. The most common clinical signs we observed were lethargy, dyspnea and wheezing, as well as fever, coughing, and then gastrointestinal illness appeared later in the course of an infection, once again mirroring the most common clinical symptoms observed in hospitalized COVID-19 patients. Interestingly, even though at 1/10 of the dose of our last study, we saw a significant increased amount of gross pulmonary lesions in SARS-CoV-2 infected cats, consisting of large multifocal areas of pulmonary hemorrhage and consolidation that were often in a cranioventral distribution, as well as more of an interstitial pattern at day 12, characterized by diffuse areas of patchy consolidation throughout the lungs, which were firm and failed to collapse. At day 4 post-inoculation, there were significant amounts of alveolar histiocytosis, or alveolar macrophages, shown here, as well as multifocal areas of inflammation and necrosis, and perivascular expansion and disruption of the vascular architecture by neutrophils and lymphocytes. We also saw a severe perivascular lymphocytic infiltrates in almost all of the infected cats at day 4, as well as several of the cats exhibiting necrotizing bronchopneumonia as well. Now, unique to the Delta variant, we saw a marked degree of peribronchiolar and perivascular edema, which may suggest that we have a more acute vascular injury with the Delta variant compared to the wild-type virus. At day 12, alveolar macrophages or alveolar histiocytosis was less pronounced, but we still had quite a significant degree of interstitial inflammation, pulmonary hemorrhage, and pulmonary edema in the lungs of infected cats. And there was still a great degree of perivascular edema and perivascular inflammation in these cats as well, although it was to a lesser degree than in day 4, indicating that these cats may have been recovering at this point. Lung histopathology was quantified by a board-certified pathologist blinded to study groups, our very own Dr. Jerry Ritchie, and revealed that pulmonary pathology was significantly elevated in all COVID-2 infected cats, especially at day 4 post-inoculation, although no significant difference was really observed between inoculation route. When we look at individual pathologic features, alveolar inflammation, alveolar histiocytosis, and peribronchial inflammation were significantly elevated at day 4 post-inoculation, as well as perivascular lymphocytic infiltrates, vasculitis, and smooth muscle hyperplasia. Overall, the degree of pulmonary pathology was less at day 12 than at day 4, but the route of infection does not appear to affect pulmonary pathology. Our molecular analyses to look at viral infection kinetics are still pending, and we hope to have some of that data here pretty soon. In summary, our feline model for SARS-CoV-2 infection induces significant clinical disease and inflammatory lesions that are analogous to COVID-19 patients. And this feline model can be optimized to suit a wide variety of translational applications, especially considering that cats frequently exhibit the same comorbidities that exacerbate COVID-19 in humans, such as hypertension, diabetes, renal disease, and obesity. I would like to thank the Oklahoma Center of Respiratory Infectious Diseases, and this research is funded by NIH through Oak Ridge. My collaborators, of course, lab manager, my students, and all the staff in the OSU College of Veterinary Medicine, and also to OSU Interact for having me here today. Be happy to take any questions.
A fantastic talk, Greg. Just questions. Have you had a chance to characterize the immune cells that are infiltrating into the lungs of this cat at different stages of infection?
The question was, have we had a chance to characterize the immune infiltrating, or the immune infiltrate in the lungs? We've started to look at that right now. So far, we've done immunohistochemistry to look at different upregulation of different inflammatory pathways, and I don't have that data. I was a little pressed for time, but we do actually see a marked upregulation at day 4 in P38, MAP kinase, and NF-kappa B in immune cells within the lungs. We have not done immunophenotype analyses quite yet, but we're still working on that.
So one of the features in the lungs is, at least when I was working with the SARS-CoV-1 and MERS-CoV, edema was quite common, and I'm not observing as much in the SARS-CoV-2 infected mouse model systems. I see here there is a less edema than expected, the previous one. Do you see that same consistency, or do you think it could be there at different stages of infection?
That's a great question. So the question was, do we see edema in this new stage of infection? And the answer is, pulmonary edema was less pronounced with the Delta variant than it was with the wild type virus, for sure. It was actually kind of surprising. The Delta variant induced more of an immune inflammatory infiltrate, so a cellular infiltrate, and perivascular edema, but pulmonary edema or accumulation of that fluid within the lungs was really not something we appreciated. It was pretty surprising.
Thank you, Dr. Miller. We are going to wrap up because we are running a little bit behind. Thank you.
Thank you. Thank you.
Okay, we are going to wrap up this session with Dr. Terrell Conway from, he's the Regents Professor and Department Head of the College of Art and Science. And welcome.
Well, good morning. Thank you to the organizers. Thank you, Dr. Beutler, for joining us today. I'm going to tell you about an idea. It's called the restaurant hypothesis of intestinal colonization. The research is founded on basic ecological principles. So David Tillman in the 70s and 80s was studying diatoms, and what he discovered was that no two species could coexist when competing for a single nutrient, but he could get two species to coexist when competing for 2 nutrients. And so his experiments were with diatoms and phosphate and silicate. He later expanded his theory of competition for resources to grasslands. In the meantime, Rolf Frieder in the 1970s and 80s was applying these basic principles to colonization of the intestine. And what came to be known later as Frieder's nutrient niche hypothesis. Rolf said that niches in the intestine are defined by nutrient availability, and that nutrients are, and this was an assumption of his theory, that nutrients in the intestine are mixed and equally available to all of the bacteria in the intestine that are competing for those nutrients. And in order to coexist, each species would utilize a limiting nutrient better than all of the other species. Now, colonization resistance would occur when a niche is occupied by an organism that's removing those nutrients and making them unavailable. In that situation, ingested microbes that don't find a niche, don't find the nutrients they need to grow, they would pass on through and fail to colonize. So in the 1990s, my students and I were happily discovering new metabolic pathways in E. coli and characterizing them. And one day I got a phone call from this guy, Paul Cohen. And he told me that my favorite pathway, it's called the Entner-Duderoff pathway, it's named after the people who discovered it. He told me that he thought that pathway was required for E. coli to colonize the mouse intestine. He had developed a mouse model of colonization. And he had a mutation in that pathway. And we sent him a good copy of the gene to complement the mutant, and sure enough, it turned out that he was right. The Entner-Duderoff pathway is required by E. coli to colonize. And so I remember asking Paul, well, what does E. coli grow on when it colonizes the intestine? He said, well, I don't know. So we decided to find out, and we worked together to the end of his life earlier this year. We were funded by the NIH for these studies. He worked at the University of Rhode Island, and I worked here in Oklahoma. And over the years, we discovered a number of principles by which E. coli colonizes the large intestine. First of all, E. coli doesn't grow on intestinal contents or on feces, but it grows great on mucus. And we would prepare mucus from mice and make little cultures in the laboratory to prove that. We learned that E. coli had to penetrate the mucus layer in order to colonize the intestine. Mutants that failed to penetrate would pass on through. We found that E. coli was in what appeared to be mixed biofilms, and what you're seeing here is a fluorescence in situ hybridization micrograph, which E. coli is stained red, and all of the other bacteria, primarily anaerobes, are stained in green. And you can see that E. coli is randomly distributed within what appears to be a mixed biofilm. So E. coli is growing in the mucus layer of the mucosum. So what is mucus? It's about 50% polysaccharide. Half of that is mucin, which is pretty clearly defined. And the rest of it's a very complex mixture of glycoproteins and glycolipids. It turns out that E. coli does not have the machinery to degrade complex polysaccharides. That's the role of the anaerobes in an intestinal microbiome community. E. coli only grows on simple sugars. Oh, too far. There we go. So this idea is illustrated here, in which the anaerobes degrade the complex polysaccharides that are available primarily from mucus, but also from undigested dietary fibers, epithelial cell debris, and as the extracellular polysaccharide hydrolases are excreted by the anaerobes, they're cutting up these complex polysaccharides, and preferentially they take up oligosaccharides, 4 or 5 sugar units, and they leave the smaller ones behind. And that's what E. coli grows on. So this is, in one respect, a symbiosis in which the anaerobes are feeding the E. coli. And this was later proven by Justin Sonnenberg and his colleagues. So the idea is that different niches occupied by E. coli are distinguished by sugars that are released by anaerobes. So as we thought about the Frieder hypothesis, in which nutrients are all mixed and available to all the organisms competing for them, we realized we had a number of experimental observations that just simply didn't fit that model. So Paul Cohen came up with the restaurant hypothesis, in which facultative anaerobes like E. coli are consuming nutrients that are provided by polysaccharide degrading anaerobes. This would occur in mixed biofilms or restaurants. So returning to the question, what does E. coli grow on in the intestine? We actually ran thousands of these mouse experiments in which we systematically knocked out one catabolic pathway in E. coli at a time. And we would compete them by growing them together in mice or feeding them together in mice. So you have a wild type that can grow on the long list of things that E. coli is able to grow on. And you have a mutant that's able to grow on all of those things except for one. And in the experiment shown on the left here, we have a mutant that cannot grow on N-acetylglucosamine. And it grows equally well with the wild type when they're fed together to the same mice. And we say in this case that this strain of E. coli does not grow on N-acetylglucosamine. In another genetic background, and this is actually the one labeled MG, that's MG1655, the prototypical E. coli K12 strain. This one does indeed use N-acetylglucosamine. So we know that N-acetylglucosamine is present in the intestine. We've measured it. We know that some strains use it and some strains don't. And so we set about looking at the sugar utilization profiles of a number of different strains, and we're showing you here 3 human commensals and 3 human E. coli pathogens. And you can see that if you just kind of scan this page quickly, you'll see that the different E. coli biotypes each use a different subset of the entire list of things that E. coli can grow on. And so this could begin to explain how there is a succession of strains in humans, how E. coli pathogens become invaders and go in and colonize in a system that's already colonized with different strains of E. coli. So it's really counterintuitive that different E. coli biotypes would grow on different nutrients. And the reason is because they have the same core genome, they have the same core housekeeping or metabolic functions. In vitro, they all use the same nutrients, and in fact, when you feed them in a mixture, they all grow on those nutrients in the same order. So phenotypically in a lab, they're the same. But in vivo, each one is using a different subset of those nutrients. So according to Frieder's hypothesis, these nutrients would be mixed and equally available to all, and so that simply doesn't explain how you can get different strains of E. coli to co-colonize the intestine. So we wanted to ask the question, for this strain that is not growing on N-acetylglucosamine or ribose or fucose or some of these other sugars, is it because they don't see them, they're in a different niche? Or is it because they just can't grow on those when they're in the intestine? So to answer that question, we used an E. coli K12, that's the MG strain, which is unable to use or grow on ribose. We compete it with the wild type, and we get the result that you expect. They co-colonize, which is saying that that E. coli K12 does not use ribose. So the question is it because it's in the intestine or it simply doesn't see that sugar in its niche? And the answer was obtained by putting ribose in the drinking water. So we're now saturating the intestine with ribose, and we're asking, is there a competitive advantage to the wild type that's able to use ribose? And the answer is yes. And that's shown graphically at the bottom here with the red bars. This is a competitive index, and we've done the same thing with several other sugars, including galactose. And so some strains see some sugars and other strains see other sugars. Now, when 2 strains are competing for the same niche, one invariably displaces the other. And in these experiments, these are the same wild-type organisms, and they're just differentially marked with antibiotic resistance markers that do not impact their ability to colonize. And so we pre-colonize with the strain, and then come in at day 10 with the same strain, and it is eliminated from the intestine. So that's what colonization resistance looks like. And that same is true for all of the different E. coli strains that we have tested. So mice behave just like humans in terms of colonization resistance. And we all know this is true, right? If we were to have contaminated hamburger for lunch, and we hope that doesn't happen, but if it were to happen, some of us would be susceptible and some of us would be resistant. And so there's always an attack rate for these pathogens. We think that's based on nutrient availability. Now, different E. coli biotypes are each able to overcome colonization resistance. And again, we've shown this with multiple times. This is true of E. coli pathogens and human commensal strains alike. So the postulate here is that one the pre-fed strain, is leaving nutrients available for the invading strain, that in this case is fed to mice at day 10, to grow up and colonize or co-colonize with the other strain. So here's a summary of evidence for the restaurant hypothesis. So first of all, different strains overcome colonization resistance to co-occupy the intestine. Different strains appear to use different nutrients to colonize, despite being able to use the same nutrients in vivo. Strains that don't use certain nutrients can use those nutrients when they're added to the drinking water. And I didn't show you the evidence for the others, but at any rate, here's the model. We call this the restaurant hypothesis. Now, they're not mutually exclusive, the Frieder hypothesis, where nutrients are equally mixed, and the restaurant hypothesis, in which different biotypes are going to different niches in which different sugars are available. What we think is going on here is that when you ingest a pathogen, your resident E. coli, an average human is colonized with 5 different biotypes of commensal E. coli, the good ones. Now you've ingested E. coli 0157, for example, the bad one. It initially has to compete for very limiting nutrients because you're already colonized with E. coli. If that strain is able to find the nutrients it needs to stay resident for just a brief period of time, it can then enter its own restaurant and it will be stably colonized. So that's what we think is going on. In invaders, this would be the new human commensal strain coming in or a pathogen, competes initially in a Frieder-like niche and then enters a restaurant in which it remains resident, and if it's a pathogen, it goes on to make Shiga toxin and cause disease symptoms. So we still don't know what a restaurant looks like. We don't know what the biogeography is. We know E. coli is red. There are very few experiments that tell us who are the green ones here. We use a universal probe, but there are a few fish experiments in which up to a dozen different colored probes are used to find different species, but they really haven't defined what the biogeography looks like yet. Ultimately, we want to put a label, a name on each of these cells in these micrographs and ask, what are they doing? We don't know in this micrograph if these are the same anaerobes surrounding a single E. coli. Where we have more than one E. coli in a little area, we don't know if they're surrounded by the same anaerobes or different ones. So that's where we are. We need to learn what it takes for E. coli to penetrate this mixed biofilm and enter into its restaurant. And that's what I brought to talk about today. Thank you.
Okay. Terrell, that was a great talk. Loved it. That was fantastic. The hypothesis and everything. I wanted to ask you, when you made your early mutants, you were knocking out the ability of the organism to consume certain types of carbon and energy sources. Well, since you're inactivating a gene, are you also inactivating the ability of other genes to function normally? And did you look into that when you looked at your mutant studies?
I think the question is, when we introduce a genetic mutation, are there unintended consequences in terms of phenotypes?
Correct.
Absolutely. You know, one of the great things about having so much experience in studying metabolism was we had learned what kinds of mutations you can make without a bad alternative phenotype. And there are sensible ways to knock pathways out. You never want to accumulate a sugar phosphate. It causes bacteria to lyse frequently, and we still don't know why. But what we always did was test the growth of this strain. I can't use that sugar, but we would test growth in the presence of that sugar. And that typically told us whether or not we had a good mutant or a bad one.
Thank you so much. We are going to take a really small break, 10 minutes, before our keynote speaker. Thank you. Thank you, Dr. Weeks.
Absolutely. So very nice to meet you.
You're right. Nice to meet you.
If we put this in presentation mode, this is right here is your best timer. So does that work okay for you?
Yeah.
Okay.
That's good. Thank you.
Okay. And then let me go ahead and I'll go over this with you while you're here. The pointer will be turned on. If you need a pointer, you got to drive it down here on the mouse pad, and then you can use this. This is forward. This is back. Don't use that one because it's so small it won't do anything for you.
Thank you.
So. Yeah, I think that yes, Dr. Vespas will start back at 10 after. So we have about 5 minutes or so left in our break. Did you all get a picture? Can I take one of you two and then is that okay? There we go. Okay, fair enough. I'm going to turn the other way. Thank you for all being here and for being kind enough to do that.
Oh, well, thank you.
Do you need a bottle of water or anything of that nature?
No, I'm fine.
Okay.
And, I just told him that
get questions, send the line channel.
Yes, yes. And
All
so we do have the potential to get questions from online, and I will ask those. I will probably just bring our mic here, just to the edge of the stage where I'm not necessarily in the live view. Ask the question from online, and then answer it from
8% of our audience actually from Europe.
8%. All right.
That's 1/10 of the audience. We can prioritize them for the questions.
Yes. Yes. I was encouraged to see the students must have been hanging out in the lobby until right before you, because they all came in a
Which, I was thinking that
so.
if you want, you can ask them to spread out. We don't care.
I think we're okay.
Yeah.
I think we're okay.
And the online audience knows that we are starting in 10 minutes.
Yes, yeah, it should have. It should have.
Who controls whether it's showing on the screen?
They do at the back of the house.
Okay.
And so I will come up. We have some videos that will play. I will give your bio.
Okay.
And I hope it's the bio you wanted. It's the one they sent me, so I hope that's all right.
My people send it?
Yes.
Then it's probably
Yes, sir.
fine.
Yes, they do.
And they control the mic,
They
too.
do. Good morning again. We're going to go ahead and get started shortly. We appreciate the patience of our virtual audience as we've tried to stay pretty close to our timeline, but we have had to make some adjustments there. So before we begin, I want to do a few housekeeping announcements. Immediately after Dr. Beutler's presentation today, we will have lunch. We encourage our students that will be joining us to travel to the second floor, to the lounge there. You can use the elevator near the registration desk to do that. And the remainder of us will be in the lobby, of course. So join us for lunch. We're interested in that. To begin with this afternoon, we have some opening remarks by some of our OSU leadership. And we'll go ahead and get that queued up, if we will.
And research that supports human and veterinary medicine is more important to society now than ever before. Interact exemplifies this mission by bringing together a wide range of disciplines from across the university to foster team science. This is evidenced by the diverse presentations from various OSU faculty members today. OSU is committed to One Health research and to collaboration, which is key to the discovery of new therapeutics and diagnostic products. Congratulations to our accomplished faculty for presenting such research today. Finally, OSU is immensely proud to host Nobel laureate Dr. Beutler. We're fortunate to learn of his scientific discoveries and advancements that have reshaped our understanding of critical immunological pathways. Thank you again, and go Pokes.
Congratulations to the Interact team led by Dr. Ashish Ranjan on organizing this symposium. In the last year, the Institute has made significant progress to unite faculty across OSU and other institutions to promote interdisciplinary One Health-related research. I extend my best wishes to the Institute as we move forward in the coming years. A very special welcome to our keynote speaker, Nobel laureate Dr. Bruce Beutler. His impactful research contributions to the field of immunology are inspiring and will motivate our faculty and students to conduct translational research in diseases that affect both animals and humans. I would also like to welcome all of our presenters today and thank them for supporting OSU's research mission.
Congratulate the Interact members and its director, Dr. Ashish Ranjan, for organizing this symposium on the OSU campus. We're highly privileged to hear all the speakers. OSU is especially honored to host Nobel laureate Dr. Beutler. We're fortunate to have researchers who are engaged in bench-to-clinic research, and listening to him will surely inspire them to do more cutting-edge research. The range of speakers and diversity of topics being discussed today is amazing. Congratulations again to the Interact team.
Thank you all for joining today. We are truly honored by your presence. Considering the ever-evolving COVID situation, planning this symposium was a challenging affair for us. I commend the Interact Organizing Committee, led by Drs. Brandau and Dr. Jones, in helping execute the symposium objectives. By adopting a hybrid format, today we are streaming the symposium worldwide, while also allowing in-person attendance at the McKnight Center. I would especially like to thank the moderators for helping run the various sessions seamlessly. As you may have discovered by now, this symposium is featuring eminent speakers from the entire university, including from the colleges of science, engineering, health sciences, and veterinary medicine. Interact's mission is to promote One Health research and conduct parallel investigations in humans and animals. And the various presentations today beautifully reflect those goals. Interact has quickly emerged as an attractive platform to forge new collaborative partnerships. For example, we recently inked a Memorandum of Understanding with the United Kingdom's Humanimal Trust to support collaborative research. Please also be on the lookout for the new grant announcement from Interact that will support university-wide research on various interdisciplinary topics. Finally, I would like to take this opportunity to sincerely thank our keynote speaker, Dr. Bruce Beutler. Dr. Beutler, we are truly grateful and honored to host you. Your presence has inspired our researchers, and we hope you enjoy the breadth of talks today. Thank you again, and over to you, Dr. Biggs.
Thank you, Dr. Ranjan. We definitely echo those sentiments as we are here today advancing One Health. We thank each of you for joining us. We would like to recognize some additional OSU leadership that is here with us today. I believe I've seen Dean Tom Kuhn from the Division of Agricultural Sciences and Natural Resources, as well as Dean Glenn Kreutz from the College of Arts and Sciences, and of course, Dean Carlos Risco is here as well. And I'm sure Dean Risco will make sure if I've missed anyone that we will get them recognized later on today. So with that, we also want to take, again, a special note to our international attendees. Dr. Ranjan took a look at our statistics over the break, and nearly 10% of our virtual audience is joining us from international locations. And in particular, we would like to recognize our collaborators in the United Kingdom as well as France. So we hope to grow that audience as we've continued with these Interact symposiums, and hope you will join us in the future. It's indeed my privilege this morning to introduce Nobel laureate Dr. Bruce Beutler. Dr. Beutler, of course, won the Nobel Prize in Physiology or Medicine in 2011. Dr. Beutler is currently a Regental Professor, Raymond and Ellen Willey Distinguished Chair in Cancer Research at the University of Texas Southwestern Medical Center in Dallas. He also serves as the director of the Center for Genetics of Host Defense. Dr. Beutler received his undergraduate degree from the University of California at San Diego and his MD degree from the University of Chicago. After his residency and postdoctoral fellowship, he started as an assistant professor at the Rockefeller University. In this role, he isolated mouse tumor necrosis factor and was first to recognize its role in inflammatory response. Later, at the University of Texas Southwestern, he leveraged recombinant inhibitors of TNF to enhance treatments of rheumatoid arthritis and other inflammatory diseases. His pioneering contributions on using TNF to identify the receptor for bacterial lipopolysaccharide has played a crucial role in signatures of infection, especially those involving toll-like receptors. In addition to the Nobel Prize, he has been awarded the Shaw Prize, the Albany Medical Center Prize in Medicine and Biomedical Research, election to the National Academy of Science and Institute of Medicine, the Frederick B. Bang Award, the Balzan Prize, the Grand Prix Charles Leopold Mayer, the William B. Coley Award, and the Robert Koch Prize, amongst other honors. Dr. Beutler is undoubtedly a pioneer in translational medicine, and it is our extreme privilege to have him here today.
Well, thank you. Thank you very much. You've all been very hospitable since I came here, and it's been a really enjoyable visit for me, especially listening to many of the talks I heard before. And I'll try to add to those, starting by telling you about how I started with an interest in innate immunity. I was drawn then to using forward genetics to try to understand things as I do now. And from there, how we accelerated the process of starting with phenotype and finding causative mutations. Then, most recently, how we've used the platform that we've developed to do that for purposes that are scientifically interesting and also may have practical value. We try to find mice that are strongly resistant to ordinarily lethal cancers and to know immediately, in a genetic sense at least, why that is. As you've heard, very early in my career, I isolated tumor necrosis factor using an unconventional kind of assay, and because of the way I isolated TNF, I had a sense that it was involved in inflammation and in shock caused by LPS, which was a very powerful TNF inducer. From that point, I wanted to understand how LPS could signal, because I realized that it wasn't known at that time just how we recognize microbes after they breach host barriers and come to be macroscopic infections. Within the first minutes or so, LPS will elicit a response, and it was known even at that time that this was important to overcoming an infection. I used TNF as a readout of my studies of LPS signaling. And by the year 1990, not too much was known about how LPS could signal, other than the fact that it would bind to a leucine-rich repeat protein called CD14 that was anchored to the surface of membranes by a GPI tether. It had no cytoplasmic domain and couldn't signal across the membrane, so it was postulated that there must be another component to the receptor complex that would cross the membrane and elicit a response. The nature of that response wasn't known, but where TNF was concerned, it was known that it could activate NF-kappa B. NF-kappa B would translocate to the nucleus and drive transcription of the TNF gene. The TNF mRNA, once produced, was in an inaccessible form, but it too would be rendered accessible by a signal from this same receptor, leading to translation of the mRNA, then processing of the protein, secretion of the protein, and then TNF would do all that it could do. I gradually became obsessed by this receptor and wanted to find it at any cost, by any means. I tried at first to purify it, then to use expression cDNA cloning, and none of these methods worked. Ultimately, classical genetics was required to track down the receptor. It happened that there were two substrains of mice, in each case with control strains, that were highly refractory to LPS, the controls being sensitive. These had arisen as a result of spontaneous mutations that had become fixed in populations early on as the mice were inbred. And one of the strains was C3H-HEJ. This had been known since 1965, that these mice were refractory to LPS. By 1980, it was shown that the animals were highly susceptible to authentic gram-negative bacteria like Salmonella typhimurium. The other strain was identified later in 1978 by Coutinho and Mayo. It was a C57 Black 10 SCCR mouse, and SCSN was taken as a control. Although these mice were really quite distant from C3H strains, it was demonstrated that you could cross the two strains, and while the mutation had recessive effects, in each case the offspring were refractory to LPS, indicating that allelic defects were involved. To find the source of a phenotype in those days, it was a very difficult and long-term kind of process. One would first map the source of the phenotype to a chromosome, which had already been done. In the case of C3H-HEJ mice, it was known that chromosome 4 locus was important, but where on chromosome 4 really there was very little definition. About half the chromosome was still a candidate. So we had to map to very high resolution on thousands of meiosis, where we took refractory mice, crossed them to a different wild-type strain, back-crossed, and then phenotyped all of the progeny and genotyped for markers up and down the chromosome. Ultimately, we confined the mutation to a position between markers B and 83.3, which we had isolated ourselves in the interim, and we built a contig of bacterial artificial chromosomes across that entire interval and began searching for genes that were there, because in those days the gene content of the mouse chromosome 4 was mostly unknown. We found only a lot of pseudogenes for a long period of time, and finally, after about 5 years, we identified the one authentic gene that was completely contained within this interval. It was a member of the toll-like receptor family, and it had no known function. Toll-like receptor 4 was exciting from the very night that we saw it in the contig. It was an outstanding candidate, although one could make up a story about almost any of the others, to be very honest about it. This one was really an excellent candidate because it too was a leucine-rich repeat protein, and we thought maybe the LPS is transferred from CD14 to this membrane-spanning protein, and that elicits a response. It did cross the membrane, and on the cytoplasmic side, there was homology between TLR4 and the interleukin-1 receptor. This was a receptor that had immunoglobulin-like ectodomain repeats, and yet it bound to a protein that had inflammatory effects, and it was known that activating this receptor could activate NF-kappa B. There was also a story from the namesake of the family, the toll protein of Drosophila melanogaster, from the Hoffman lab, where it had been shown that toll, which was known to be involved in dorsal ventral patterning of the embryo, also was required in adult flies for defense against fungal infection. If you knock out toll or its protein ligand Spätzle, flies are susceptible to injection of Aspergillus fumigatus, for example, and that can be seen with fungal hyphae sprouting from the corpse of a fly. I mentioned to you already that the gene we were looking for was known to be required to defend mice against E. coli infection, Salmonella typhimurium, and so forth, so it seemed we might have a parallel story. We dropped sequencing of other candidates in the contig, and we focused only on this one and quickly found by shotgun sequencing long cDNAs from HEJ and HEN strains that there was a single nucleotide difference which translated to a single amino acid difference in the cytoplasmic domain of the protein, where normally there was a proline in the sensitive strain, there was a histidine in the refractory strain. That would not have been enough to claim that this was the causative mutation, however, because we didn't really know for a fact that it was deleterious, conserved though it was. Remember, though, we had another strain to look at, another allele, and when we sequenced TLR4 of the C57 Black10 SCCR strain, we first found that none of the exons were present, and there was no RNA coding for TLR4 in C57 Black10 SCCR mice. With a bit more work, we found that this was a 74,000 base pair deletion, the limits of which we defined by making primers farther and farther out from the gene, and this definitely was a deleterious allele, and with that we felt quite confident that we had identified the product of the so-called LPS locus. This discovery was exciting again from the very first day because we saw that there were paralogs of toll-like receptors in mammals. At the time there were 5 of them. Later we found several more, and now we know of 12 in the mouse, 10 in the human, 13 in both species combined. And by directly targeting these genes, mostly in the laboratory of Shizuo Akira, it was determined that each of them recognized a different inducer of TNF from different microbial species. These were molecules like lipopeptides that were present in both gram-negative and gram-positive bacteria, nucleic acids that were characteristic of all microbes, double-stranded RNA, for example, single-stranded or double-stranded DNA. Flagellin would bind to TLR5, and I've told you about LPS, which bound to TLR4 as well as another protein which was found a bit later by Miyake and colleagues, a molecule called MD2 that makes a hydrophobic pocket within which LPS sits. It was also clear that parts of these molecules were conserved across the tree of life, and not only invertebrates and vertebrates, exemplified here by a human, but also plants were dependent on these proteins to defend themselves against a wide variety of pathogens. This was truly an ancient aspect of innate immunity, a sensory mechanism that was used by almost all multicellular life forms. While this had taken me 5 years, I'd become thoroughly immersed in it, and I loved the idea of going from a phenotype to finding a mutation that was responsible for it. But first of all, there weren't so many informative phenotypes around. Many of them had been spoken for, and people had cloned them or were on their way to doing so. It took too long, frankly. It took years to track down this one important phenotype, and I wanted to accelerate the process, the goal being to find critical parts of a machine which one could then use other methods, biochemical approaches, for example, to try to understand how they operate together. And I had in mind the TLR signaling pathway, and we did make a lot of progress with that using forward genetics. The idea then was to make new phenotype de novo on a very conserved homozygous background in mice on an inbred strain, and for this purpose we use the black 6 strain. One does this by injecting an alkylating agent, ethyl nitrosourea, into male mice, whereon it goes to the spermatogonia and it creates single base pair changes at random across the genome. These changes are transmitted to G1 animals, and we take G1 males as the founders of pedigrees. They're heterozygous for these mutations, which must therefore be viable at least in the heterozygous state. And we breed these again to black 6 females to make G2 daughters. The G2 daughters are crossed to their father within each pedigree, and one then has G3 animals where you can have homozygosity for the mutations that were first fixed in the germline in the G1 mice. Over a period of about 11 years working at Scripps Research Institute, I dedicated myself to this, and we made and solved about 200 phenotypes, some of them visible, like the ones shown here for illustrative purposes, but many of them having to do with innate immune function. What we learned from this exercise was that all of the phenotypes we solved came from coding change. They were either missense or nonsense mutations, or they affected splicing. There were no remote intergenic mutations responsible for phenotype, and that's not only our experience, but it's the experience of the whole field. It's extremely rare that you find a phenotype coming from a remote intergenic mutation. To take just one example, we would look in those days at all the G3 mice. We didn't really care which pedigree they had come from. We would look in screening to see if they were exceptional in some way or another. One of our screens was to give LPS and to look for TNF-alpha, as we had done with C3H HEJ mice in our mapping studies. And here's an example of a G3 that was an extreme outlier. It made a very poor TNF response to LPS. When we crossed it to mice of the same strain, the phenotype disappeared because it was recessive. When we crossed these animals back to the founder, we saw responder and non-responder populations in equal proportion. This was something we could work with. We made an extensive stock of homozygotes. We outcrossed it, we backcrossed it, we mapped it, and eventually we found it. It was a mutation that we called LPS2. It was later known as TRIF or TCAM1, and this was the very first hit that we found in our screen for innate immune defects. Later we found about 20 others, and together they give us a quite strong view of how the pathway from TLR4 to activation of TNF works and what must happen to allow that. You might imagine that false positives were common when we did things this way. That outlier could have been just a fluke, maybe a bad ELISA assay. Actually, it was a bioassay, even more prone to error, that we used in those days. Also, false negatives were common. Only extreme outliers could be found and mapped using this approach. And let me explain why that's the case. You might have an assay, doesn't matter what it is. If it's a quantitative assay and you do it pedigree by pedigree, which is really the better way to do it, using larger pedigrees than we used in those days, if you find a couple of outliers like this, you can be sure it wasn't a fluke and that this is really statistically significant, and you say, let's go for it. That's something we can track down and positionally clone. But if these two outliers were more subtle, everything else being the same, if they were like this, you would look at the results from this pedigree and you'd say, there's really nothing here we can work with. We better go on and find another pedigree that has something more promising. If you had a magical tool, however, that told you within your pedigree there is a mutation for which these two mice are homozygous and for which all the mice shown in green points are heterozygotes and those shown in yellow are reference allele homozygotes, you would say there is something going on here. This wouldn't happen by chance alone. It's too much of a skewing of zygosity with phenotype. And these days we do have such a tool, and we usually express our results in columnar fashion, but suffice it to say that we would know immediately what the mutation causing this phenotype was. We developed this platform shortly after I moved back from Scripps to UT Southwestern, and beginning in 2013, we put it into large-scale operation, and we began positionally cloning phenotypes in real time, which is to say thousands of times faster than we'd ever been able to do before. Let me explain how the platform works. As ever, we pour mutations into the germline genome of mice using ethyl nitrosourea. We breed them to make male offspring, but these days we whole exome sequence every male mouse, and that way we know all of the mutations within coding and splicing regions that they're able to transmit to their G2 and G3 progeny. This is done using an Illumina sequencer, and we used to guess about how many coding and splicing changes there were. Actually, it was more than I would have thought. The number is very close to 60, and there's considerable variation, looking now here at thousands of whole exome sequences from G1 males. We don't like to be trying to positionally clone or identify phenotypes within mice with a very low number of mutations. Typically, we establish a cutoff of 30 mutations to be adequate, and if a mouse has 30 or more coding splicing changes, then we move forward and we breed it, and at the same time we order a panel of PCR primers that flank every mutation site in the whole exome so that we can amplify and genotype them later on and establish the zygosity of G2 and G3 descendants of this mouse. These days, we don't just make a couple of G2 daughters, we make 10 to 15 G2 daughters by harem breeding the G1 mouse, and we strive to make 50 or so G3 animals. We usually succeed at that. As mentioned, all of these animals then have to be genotyped, and that way we know what happened to all the mutations, and we know critically whether every mouse at every mutation site is homozygous, heterozygous, or reference allele homozygous for the mutation in question. This is determined using ion torrent sequencing, which is the right tool to use if you have to genotype, custom genotype thousands of different mice. And this will show us on a scale here of 0 to 100% mutant allele calls, whether you have a homozygous variant or VAR genotype, a heterozygous variant or het, or homozygous reference allele or ref genotype in the language of our computer people. These genotypes, of course, are not even looked at by humans. They're just uploaded and they reside within the computer and it codes them, and you can mouse over them if a particular mouse is in question, but we usually don't do that. VAR is light blue, het is green, ref is yellow. Some mutations turn out not to have been true, or for technical reasons they fail, and in either case they're removed from the picture by the computer. Only at this point, when a whole pedigree has been genotyped, is it submitted to a phenotyping pipeline. And in this pipeline, lots of different biological phenomena of interest to us are kept under surveillance. And we do an average of about 100 different phenotypic measurements per mouse by a crew of people who get to be very fast at phenotyping and who work day and night to do it. And we find that we can look first with non-invasive tests like body weight or motor performance on a rotorod. Then we can do rather invasive things. We can immunize the mice, and we can determine whether they have normal T-dependent and T-independent B-cell responses or allergic responses. We bleed them, and we do flow cytometry on all of the blood cells in the periphery, and we look for abnormalities of FACS in the mice. We also do a glucose tolerance test. We do measurements of blood pressure and heart rate. We x-ray the mice and look at bone density, length, morphology. We give the mice dextran sodium sulfate in an amount that's harmless to the wild type, but we look for severe colitis occurring in the G3 animals as an exception. I won't go through all of them, but I want you to know that one of the tests we began doing about 32,000 mutations ago was to screen for resistance to an ordinarily lethal melanoma, asking whether we can detect exceptions there. And I'll return to that a bit later. These days, it's the computer that brings to our attention any causative mutations. This is done by a combination of statistical computation and also machine learning assessments that are made to take account of peculiarities of the data in each of the screens that we do. Let me just show you what it looks like to us. We first would see a Manhattan plot if the computer says this is something to look at. Here, every mutation within the pedigree is shown as a dot, and they're arranged chromosome by chromosome in centromere to telomere order. And we're looking on the y-axis at the negative log of the p-value, testing the null hypothesis for each mutation that this mutation has nothing to do with the phenotype. It's unrelated to all of the phenotypic data. And usually the null hypothesis is not rejected. This is the p.05 line in pink. The red line is with Bonferroni correction, since we're looking at all these many mutations. But in one particular case, as you see in this example, there's a very strong rejection of the null hypothesis with a p-value of better than 10 to the minus 9th. If you click on that point, you see that it's a nicastrin gene, a mutation in the nicastrin gene, and the computer has already brought to a collection lots of information about the nicastrin gene, so if you didn't know it already, you would find out that this is part of the gamma secretase complex, which cuts membrane proteins within the plane of the membrane, and you would learn quite a bit about what's known of the knockout phenotype. It's homozygous lethal. And you would also see where your mutation was and what it was, V493G in this case, and it would all be interactive, so you could see what kind of motif that is. You can look also at a gene model and you find which exon it's in, if it affects splicing or is predicted to do so, that is shown to you, and what the outcome would be. All these things you used to do by hand. Also, very importantly, it'll tell you whether you've ever looked at this gene before, if you've hit it with mutations in the past, and in this case you have, and you can mouse over those and look at the mutations and see what they are if you wish. If you right-click on this dot, then you see a scatter plot showing you the data that gave you that strong p-value. This is the phenotypic performance in a T-independent B-cell response screen of homozygotes, heterozygotes, reference allele homozygotes, and wild-type mice that are included as an additional control. And obviously that wouldn't happen by chance. It's a rather extreme example. In this same pedigree, there were some mice that were hypopigmented, and that could have been from some other mutation that was present, maybe a Hermansky-Pudlak syndrome mutation, maybe something else. But the computer tells us immediately that came from the same mutation. It's also doing that. In fact, many mutations produce numerous phenotypes that are detected among the 100 or so assays that we do on the mice. and you get right away a picture of the physiological importance of the mutation. Because the knockout of this gene is homozygous lethal, we recreate the mutation to be absolutely sure about it. We use CRISPR-Cas9 to do a replacement on the very same site in the gene to make the same amino acid substitution, whereupon we see that these mice, homozygotes of that clean mutation, are the same heterozygotes with no effect. Reference allele homozygotes are normal, similar to wild type. It verifies. In fact, there were numerous screens that had to be rechecked. The visible phenotype validated as well. The other mutations that affected immunity also validated. The stringency of this determination of validation is made by the computer, and it's very strict. So there are some things that had good p-values in the past, and yet they don't verify because they're not quite up to what's required, but you can keep them in mind as likely to validate if you use a larger series. When you do this for a long period of time, as mentioned, you hit the same genes over and over again. We've looked now at close to 240,000 mutations in our screens, and here would be an example of a FACS abnormality that was seen in 7 different pedigrees involving the same gene. The computer will put this information together, making what we call a super pedigree, and it has hundreds of mutations because they come from all these different pedigrees. Most of them don't score in the screen. Some of them do a little bit because that's how it is with FACS screening. You look at hundreds of mutations and you find some things that do get to be significant when you hit them over and over again. But this one has an astronomical screen, and you have the opportunity to look at the scatter plot of all the results from the pooled pedigrees with color coding for different alleles and with the size of the dots corresponding to the likelihood of damage to the protein in question. This goes on day after day in the lab. Once the pipeline for mutagenesis has been started, it's not easy to interrupt it, so we try to keep it going and never run out of money to support it. And here you have another example of a Manhattan plot and the corresponding scatter plot. And these two would correspond to each other as well. And you see it's always a very nice looking spike, or usually so. Sometimes you have an isolated mutation on the chromosome, and there's no question of co-localization or which mutation is causative, but sometimes you do have trouble with that. Here would be 50 more examples, all very nice quality data. Here are 400 more examples, so I'm sure you can barely see it anymore, maybe on the highest resolution screen in the auditorium. But the point I want to make here is just that we would have to show you many such pages, because to this time we've finished 23,462 declarations of phenotype ascribed to particular mutations, and that means 23,462 finished positional cloning projects. What used to take years now takes no time at all. There's more than just this brute statistical computation involved. You can imagine that we would be influenced by many other things, but we try to be still unbiased as far as what literature says about mutations, for example, because literature can be wrong. Genetics should never be wrong. But the question of whether there are multiple alleles supporting the phenotype is obviously important to your own conclusion about whether something is causative or not. Is the assay reliable? What's the damage probability of the mutation? Is the gene in which the mutation landed an essential gene for life, like the nicastrin gene? If it is the case that such a gene, if mutated by a viable hypomorphic mutation, is likely to show a lot of phenotypes, more likely than something that's not essential for life. What's the quality of the data? How much scatter was there? Was a single outlier responsible for the significance? Was there co-segregation of multiple mutations in the peak that you observed? All of these things are quite important, and there are more. Humans are rather difficult, find it rather difficult to assess the relative merits of all of these different, let's say, features of the data. And yet we did try. We made a human algorithm where we accorded a point scoring system to each of those different features, and there were in all 67 of them by the time we were done thinking about how we interpret the data. And this did better than nothing in a receiver operating characteristic curve looking at sensitivity versus 1 minus specificity. The area under the curve was 0.74. A perfect diagnostic, of course, it would have a value of 1 under the curve. So it helped us, but it wasn't really the best. We therefore made a real machine learning program incorporating all of those features and a few others. And in this case, we trained the machine learning program based on the outcome of our CRISPR-Cas9 retargeting work. We retargeted something like 550 genes, and we analyzed those in a total of more than 5,000 assays that had been done in primary screening. The computer made its call, and then we checked and found out whether it was right or not. And with training, we got the area under the curve to 0.944, which is really a very good diagnostic tool. And in fact, it's so good that the computer has been given complete autonomy now in declaring causation. It's just better than we are, and we tend to be biased, and we like to let the computer make these assignments by itself. The computer can also tell us for any phenotype that we have, any mutation phenotype association, how likely it is to be verified by CRISPR-Cas9. And this goes from the Candidate Explorer score, the machine learning score plotted on this axis, versus probability of verification on this axis, looking at windows of 0.01 in the CE score, to the point that you can look at these and you can say, we barely really need to make a CRISPR on these. They're going to verify. We always do if it's a critical matter, but we wind up wasting a lot less time than we did in the old days. Of course, this platform is wonderful, and mainly we used it to create new diseases. We made more than 100 new immunologic deficiency diseases, and that were novel, I'll say. We made actually in the thousands of immune abnormalities, but more than 100 were clearly novel and were clearly true. At least they had better than a 0.95 score with the likelihood of verification. And those we published, and I find some of them very interesting, and we're going to work further with them. But at some point we decided it'd be useful to turn the platform around and try to mitigate disease. Because of course there are disease modifier loci which will mitigate disease. Could take a few examples. There are people who are homozygous for sickle mutations in the beta globin locus, and yet they never develop severe disease because they have hereditary persistence of fetal hemoglobin, which can result from mutations in a couple of different places in the genome. There have also been drugs designed based on modifier loci. For example, you might take Maraviroc, which was a drug that targets CCR5, and the original observation was that humans with a deletion in CCR5 are quite resistant to HIV, show very slow progression of disease. And there are other examples too, but very, very few of them. And we thought, why not use this system to find disease modifier loci at very high speed and really interrogate the genome at depth, using an invasive screen that one could never even think of using in humans, and that screen would be to find resistance to assuredly lethal cancers in mice. We decided to use the B16 F10 melanoma for that purpose, giving 200,000 live melanoma cells subcutaneously in mice, unless it's a bad injection, will always lead to the death of a wild-type mouse. Within a period of about 20 days, the tumors are large enough that we must euthanize the mice. In the assay, we gave that inoculum, and on days 5, 8, and 11, we also gave an anti-PD-1 antibody to dose to the mouse. And we did this knowing that it has no effect at all on the progress of that particular cancer in mice. In humans as well, with melanoma, anti-PD-1 is good in about 30% of cases, but in 70% of people, it doesn't have an influence on the course of melanoma, and we thought maybe we'll find mutations that are detectable in this sensitized screen, and it might teach us something even about why some humans are responsive and others not. The readout here was to look at the volume of the tumor by measuring with a caliper on day 13 and on day 20, and if mice showed no tumor growth at all, we would recycle them through the screen to see whether that was a bad injection or whether it was something real. After a time, we found the first of our mutations, which was a really strong effect mutation, and we named it Icebreaker to connote some kind of breakthrough, I guess. It was the name of the postdoc who found it, Ying Wang. And in this particular pedigree, there were 8 homozygotes for the mutation, and 5 of them showed no tumor growth at all, and when we recycled them, still there was no tumor growth at all. And because those mice were survivors, we were able to breed them immediately and establish a stock, a large number of homozygotes, and also heterozygotes and also reference allele mice. And we looked at these mice, 20 per group, with and without anti-PD-1 antibodies, using IgG as a control. We found, first of all, that this was a strictly recessive mutation. If you took Icebreaker mice and you didn't give them any protection with anti-PD-1, they still showed a strong resistance phenotype, but none of them were cured in this case, and some of them would go on to die, most certainly, if we let them go beyond 20 days. If you did give anti-PD-1, it was significantly better, and again, about half of them were self-curing, and half of them would go on to die. The heterozygotes and homozygotes for the reference allele, whether you gave them anti-PD-1 or not, they would all go on to die. We got even much more excited when we looked at other cancers. We found that homozygotes for Icebreaker were resistant to the Lewis lung carcinoma, also a very lethal kind of cancer in mice. And we found that with MC38, without checkpoint inhibitor in this case, the tumor would grow for a period of 6 to 8 days in homozygotes, and then it would begin to undergo involution, and it would be completely rejected, and 100% of these mice that were homozygous for this mutation were self-curing. We did, needless to say, verify that this was the causative mutation by recreating it on a clean background, and we saw exactly the same phenotype. Here we're looking with MC38, but it was the same with the other tumors as well. If a mouse cured itself of MC38 or any of the other tumors, then it was extremely resistant to inoculation with the other tumors. None of them would ever begin to grow. They would never form a tumor mass in these mice. It would be rejected very quickly. And this property of memory, among other things, led us to think that this was almost certainly immunologically based. We found, looking at tumor-infiltrating lymphocytes, that on days 8 and 10 there was a strong excess in the number of total tumor-infiltrating cells in Icebreaker tumors. And particularly there was a high number of activated CD8 T cells, CD44 high T cells. This outnumbered the number of those cells in heterozygotes or reference allele mice by a factor of about 100 on day 10. We depleted CD8 T cells with antibodies in these mice, and if you look at the red curves, you see that with anti-CD8 antibody in Icebreaker and in wild-type homozygotes, the tumor is essentially behaving equally. It completely removes this tumor resistance phenotype. Absolutely depends on CD8 T cells being present, but not CD4 cells, not NK cells. We thought then that very likely this mutation landed within a certain protein that we knew that protein is critical for life, but the protein has its own class 1 presented peptides, and we thought that perhaps we had modified one of them in such a way that in Icebreaker mice, there's no particular problem because that's seen as self, but when a tumor is introduced with the wild-type form of the peptide, the tumor gets attacked on that basis and rejected. So we knocked out, if I can call it that, the icebreaker protein in melanoma cells. And these cells were also made to express ovalbumin because at this stage we didn't know whether that hypothesis that we had was really correct or whether CD8 cells were just extremely active against anything and were acting against other neoantigens that were present in the tumor, so we wanted one to follow. What we observed was that if the icebreaker gene was knocked out in tumor cells, as shown by the green and red curves, those cells could grow aggressively and overwhelm the mouse. And the reaction to them was indeed dependent on the presence of that protein, and we assumed, therefore, was related to recognition of a single amino acid change induced by ENU in the icebreaker strain. Knowing where the mutation was, we could look at candidate peptides to be presented around that mutation site, and we made both wild-type and mutant versions of the peptide, and we then loaded antigen-presenting cells from icebreaker mice with wild-type peptide or mutant peptide and injected them intravenously into a tumor-experienced mouse. By 72 hours, you can see that if we loaded the mutant peptide, the cells are doing just fine, but the cells are being eliminated if a nonomer of the wild-type peptide has been loaded, even more if an octamer of the wild-type peptide has been loaded. The icebreaker mice are reacting against this peptide, and that's the reason they're able to reject the tumor. But I must say, and I want you to bear in mind, that among 32,000 mutations that we looked at, that's the only one that works that way. We have altered something like 5% of all predicted and 5% of all known class 1 presented peptides in the proteome with mutations, in the majority of cases destroying them actually because we've created premature stop codons or frame shift mutations or splicing errors. And it's very rare that you can get a single amino acid change to cause rejection of a tumor. Knowing what this was, then, we could make MHC tetramers, which would let us look at T cells that expressed a receptor that was reactive with that peptide. And in a tumor-cured icebreaker mouse, we could see the T cells that led to the cure. They amounted to something like 2% of all the CD8 positive T cells in the mouse. It is known that minor histocompatibility antigen differences, and this is essentially what we created with the icebreaker mutation, can be helpful in cases of leukemia where allotransplantation has been performed and you have a graft versus leukemia effect. And usually when you have graft versus leukemia, if it's restricted, it's not such a terrible problem, but it can be. It can be dangerous. You can have really fairly severe chronic GVH disease on this basis. It isn't known that this will reject solid tumors, and that's something remarkable about this one amino acid change and its candidacy as a target. It's really quite remarkable. We know it's safe. We know it rejects really aggressive solid tumors in the mouse, but would it be safe if we tried to use this as an actionable target in treating cancer, for example, in humans, by transfecting in a human TCR reactive with that peptide? Would it cause havoc in the host? We started to look at that by grafting albino black 6 skin onto icebreaker mice, and we found that it was not rejected, at least usually not. These are mice that have been growing grafts for about 3 months. We thought maybe Tregs present in the graft are mediating tolerance to this amino acid difference, and that doesn't take place in tumors. We then took nude skin and grafted it onto the mice, and that was accepted as well. And therefore, it doesn't seem to be anything involving Tregs that protects the grafts. We also transferred from tumor-experienced mice, tetramer-positive mice that is, RAG2 knockout mice and also wild-type mice with cells, and we found that this did not produce any kind of GVH disease, even though the RAG mice became well populated with tetramer-positive cells. The mice weren't harmed by it. But looking at these same mice that didn't lose weight and were looking quite healthy, if we then injected them with tumors, the presence of tetramer-positive cells would very significantly interfere with the growth of the tumor. In these experiments, we gave only 200,000 tetramer-positive cells to the recipient mice, and that's not very many. What this tells me is that we at least have some window of opportunity to try to treat tumors using what we've learned about this as a special target. Toward that end, we began cloning T-cell receptors that were reactive with the MHC tetramer, and we found the best of these was something that we call TIL3. It would react very clearly against the T-cell receptor. The T-cell receptor would react against the tetramer in TIL3, and we cloned the alpha and beta chains of that receptor, among quite a few others. We then put these into a retroviral vector, and we used that to transfect CD8 T cells, which were then given to a RAG2 deficient mouse. And after 3 weeks, we inoculated the mouse with MC38 in one flank, B16 in the other flank. And again, we observed protection against both of these tumors, this in the absence of any checkpoint inhibitor. In one case, we had cure of both of the tumors. So now we're very keen to clone a human TCR with reactivity against this target. Here's another strong resistance mutation. We call this one citation. It just barely passed the Bonferroni correction limit, but that's more a property of this screen, which gives quite a lot of variation in tumor size. On days 13 and even more so on day 20, you can see that homozygotes for this mutation will restrict the growth of melanoma. In this case, the gene that's been hit encodes a cell surface protein, and there is quite a lot known about this protein, but no previous suggestion that the protein in the normal wild-type condition restricts resistance to cancer. The mutation essentially is a knockout of the gene, and we found rather early on in direct, complete bone marrow transplantation and mixed bone marrow chimera experiments into fully irradiated recipients that even if you give half of the bone marrow from wild type and half from the mutant, and you see that that's present in the chimera, you get a full effect in restricting the growth of melanoma. We also observed by flow cytometry that both plasmacytoid dendritic cells and conventional dendritic cells are more numerous in the blood of citation homozygotes. You see that here and here, and this is true whether they have a tumor or not. It's something that's constitutively present. We see, too, that conventional dendritic cells have much higher expression of class 1 MHC, and all this made us believe that perhaps the mutation was eliciting its effects in dendritic cells. We did an experiment with melanoma transfected to express ovalbumin again, in this case because we could use OT1 cells as an indicator of the priming effect of dendritic cells in wild-type mice and in citation mice. We took dendritic cells from the draining lymph nodes of these tumors, which grew faster, of course, in wild-type than in citation, as I've tried to illustrate here. If dendritic cells were harvested from wild-type mice and used to prime the OT1 cells, which recognize the synfecal peptide of ovalbumin, you didn't get priming by day 6 following tumor inoculation, but you got very aggressive priming from the dendritic cells of citation mice. More evidence that this is really how the mutation is working. Probably it enables citation mice to respond much better to neoantigens that are presented by citation DCs. We then knocked out the gene in question just in dendritic cells using a CD11C Cre driver, and that was sufficient to closely phenocopy the germline citation mutation. There was a question still, is this something that's developmental, or what happens if you only knock it out in an adult mouse with a fully functional immune system? To address that question, we made a UBC Cre driver construct mouse where we could induce knockout of the citation gene by administering tamoxifen. In this experiment, we gave tamoxifen to the mice 2 weeks prior to tumor administration, and we documented that indeed the gene was knocked out in cells of hematopoietic origin, including dendritic cells, and we saw again the tumor resistance phenotype in its full form. This makes us feel that the protein in question here might be something that we could block for therapeutic effect using a monoclonal antibody, and we're working in that direction now. I won't have much to say about this mutation, but there is another that we found that's not quite as strong as the other two. We call it whalebone, and you can see that mice homozygous for the whalebone mutation show something like 50% or better reduction in tumor volume by day 20, but they would all go on to die from the tumor if you let them sit with the tumor long enough. Remember, in icebreaker mice, about half of the mice survive completely and are cured, and half of them are not cured and will go on later to eventually die. We made double homozygotes of these mice, and we found 100% salvage of the double homozygotes. All of them are self-curing, something we would never observe with icebreaker alone, and certainly not with whalebone alone, which means that there's synergy between the two of them. We have in all 12 mutations that cause at least some verifiable degree of resistance. And what I find just remarkable is that by putting 2 nucleotide changes into a black 6 mouse at different positions in the genome, you make that mouse completely resistant to a lethal cancer. Really just enormously surprising, and most of these mutations should be actionable, at least we can imagine ways in which they would be. So we find this to be a productive way to move forward in the future, and I think it could not be done by any other way than the platform that I've described to you. I think there should be some time for questions still. I think I kept to time, and I want just to say that in the beginning things were very difficult when it came to tracking down our very first gene, the LPS locus. Alexander Poltorak was the real hero of that effort, but I had help also from Betsy Layton, Christoph van Uffel, who began that project, and Irina Smirnova, who worked on it till the end. Now, after you get some recognition for doing one good thing, you can afford to do larger scale kind of work. Prior to COVID, this was our group as it stood in 2017. A few people have vanished in the meantime due to the difficulty of working under these conditions, but we still have quite a big group, and we still do mutagenesis, and we're making as many mutations as we can in the area I told you. The work I showed you with cancer resistance was all done by Hexin Shi and Xiaoming Zhan. And with that, I'd be happy to answer questions. Thank you.
We encourage our live audience to come forward to our mic in front of the podium to ask questions. I would like to remind our virtual audience that you're able to submit questions via email, and we'll be monitoring that really as we speak. Submit those questions to interact@okstate.edu. Again, that's interact@okstate.edu. We do have one question here in the audience. Go ahead.
Great talk, quite encouraging. So my question is related to the earlier part of the talk when you use ENU to induce mutation in the first place. And you commented or mentioned about that these are only targeted to coding region, not the non-coding region of the genome. So what's your thought in it? Why it's so specific to coding part of the genome?
It is a random mutagen, and if you look at a whole genome sequence, you'll see about 3,000, 3,500 mutations genome-wide. But the coding region is where phenotype emanates from. And the reason for that, I think, is that most genes have quite a few motifs that are required for transcription. There's enough redundancy that it's just very rare that you can have a detectable effect in any of our screens, at least, by mutating just one of those motifs. It's not absolutely unheard of. There's a case of a mouse where there was a limb deformity caused by a mutation in a cis-acting limb organizing center that was about 1 megabase away from sonic hedgehog. And it worked its effect by affecting sonic hedgehog expression. So it can happen, but 99+% of the time, if you have a phenotype, it comes from coding region if you're using a point mutagen.
Thanks.
Great talk. Thank you very much. I do have one basic immunology question. I saw that the site mutations had high MSC and PDC and CDC expression. Do we know what is the basis for this increase?
What kind of mutations did you say?
It's a citation mutation, if I'm remember
Oh,
correctly.
yes, citation. Do we know what's the basis for that increase?
Yeah.
We have only ideas about it. It's not something that anybody has ever noticed before, and I can't really say more about that.
Okay, thank you.
Yeah, good talk. I just have a very general question. It's about total-like receptors. And Blunt just recently that they also exist in plants. Is that right?
Something like them. The TIR domain exists in many cytoplasmic disease resistance genes, and you have some leucine-rich repeat proteins which don't have a TIR domain, but which have been demonstrated in Arabidopsis, for example, and in rice by someone I know very well in the case of rice, Pamela Ronald, who happens to be my second cousin.
Oh.
So I mentioned her name.
Yeah, so I was wondering what can we learn? Because you are more focused, I see your study is more about humans and maybe animal models, but what can we differentiate from plants in terms of trying to bring them together?
Well, I think that it just shows that there is a system that's ancestrally conserved to that degree. It's something like 1 billion years ago, this mechanism was seized upon, and it was so effective in dealing with microbial infection that it was kept even to the present day in all across the tree of life.
Oh, thank you.
That was awesome. So you mentioned this interesting thing that you've generated about 100 phenotypes, or 100, I guess, genotype-phenotype matches that are not known in nature, where you have some mutation you've made with a phenotype that was unknown before. Is that right?
Yes, well, yes, at least in the case of these immune phenotypes, they are novel. Nobody has noticed that if you mutate such and such gene, it wipes out all your T cells. Hasn't been observed in humans.
Yeah, so I would like you to speculate on why that is, with this huge experiment of billions of us going on all the time, and presuming that maybe these mutations are not embryonic lethal. Why don't we ever detect them in humans, or why haven't we? Is the phenotype not severe enough? Or?
In some cases, there might be redundancy that just protects a human so it doesn't happen. But in some cases, I know for a fact people just didn't notice it yet, and they're bound to find it sooner or later. There's still more to discover.
As our next individual comes up to ask a question, we want to remind our virtual audience you're able to submit questions at interact@okstate.edu.
Thank you. Great talk. I'm interested in behavior, and I wanted to ask if with this great library and this number of animals with all these mutations, were you able to observe any modifications in the behavior of these animals or links to psychiatric disorders?
Yes, well, we see behavioral disorders that are sometimes very interesting and haven't been adequately researched yet. Quite a few years ago, we found a mutation called possum. When you handle mice, you usually scruff them, and you then gently put them down. And in one particular case, one of the animal handlers scruffed a mouse and put it down, and it appeared to be dead. It stopped breathing. And she watched it for a little while, and it started breathing again, and it remained in a kind of a death-like trance for a period of, let's say, about 5 minutes. And then it got up and trundled away, and it was fine. And then she did it again, exactly the same result. We named that possum, and we mapped it. This was a little bit in the older times, and we found that it was a mutation in SCN10A, which is a voltage-gated sodium channel. And the nature of the mutation is such that the channel kind of gets stuck in the open position, and the cells of the dorsal root ganglia are flooded with sodium. Very interesting point. This is a complex behavior, also involves bradycardia, but there's no change in brain waves in the mouse. It's conscious while it's lying there. And there is no expression of this protein in the central nervous system. So how do you get such a complex behavior from something that's not even in the brain? That's what puzzles me. And it's only scruffing that does it as well. You can't do it by pricking the mouse's foot with a pin or by pinching the mouse elsewhere. Other noxious stimuli don't seem to evoke this at all. So I wish someone would work more on that, actually. Other mutations we really have screened for with someone named Maria Charroux in UT Southwestern. A screen for autism-related phenotypes has been carried out. Separating infant mice from their mothers, you get normally kind of an ultrasonic cry that they emit. And this can be measured with a microphone suspended over mice in a kind of nice array. So you can monitor on a large scale if there are abnormalities in that behavior. And she did. And she found a mutation in KDM5A. And this she has published not so long ago. There are people with language development problems who also have mutations in KDM5A. And that was discovered subsequent to the mouse discovery by Maria. So I love developmental and behavioral phenotypes as well. And some of them are just so interesting, I wish I had another lifetime to work on those.
We recognize we may have a delay with our virtual audience, and so we just want to assure them if you have questions and you're joining us virtually, you may submit those to the Interact email, and we will do our best at the conclusion of the symposium to try to get those addressed for you in the next few days.
Great talk, thank you. I'm curious, for the third case, you described that 50% of mice were resistant, but 50% survived, were not survivals, the other way around. So I'm wondering, what was different in those 50%? And I'm also curious to learn whether you're planning to look at other cancers than melanoma?
Yes, it's a good question. You think the mice are about as genetically identical in that group as they can be, but they're not 100% identical, so we have to bear that in mind. It could be epigenetic differences between the mice. It could be just something stochastic. And I don't know, sometimes you have a certain screen or a biological phenomenon and it seems to be balanced on a knife edge, and you have survivors and non-survivors, even if you make things as identical as you possibly can. We are looking at other cancers too. Some of them we created in our lab by making P53 knockout mice, which spontaneously develop a variety of tumors. We also had folliculin mutant mice that came up in our screen, and those too developed tumors. And we were sort of checking a lot of them because we like to have a large stable of them to look at.
All right, I think, Dr. Beutler, this will be our final question.
Translation. I was curious with the icebreaker mutation. From my understanding, there's an overexpression of CD8 T cells. I know that is essential for the adaptive immune system and for actual viral infections. So have you done any trials looking at using the icebreaker mutation for viral infection, like with influenza?
We didn't do that. It was something that we considered doing for a time, but after we found that the reactivity of icebreaker was based just on that one peptide that is mutated, and it would react against the wild type form that it had never seen and did so as though it was foreign, then we lost enthusiasm for going that direction. So I truly believe it's really all based on the T cell reactivity against that one amino acid. Wouldn't probably make a difference in its general antiviral responses.
Okay, thank you.
Okay.
Dr. Beutler, thank you so much for being here. Let's give him a kind round of applause. On behalf of the Interact team, Oklahoma State University, we can't thank you enough again for joining us. I believe you'll be visiting with maybe our students a bit over lunch. We are running a bit late, particularly for our virtual audience. You may have noticed that we're running about 30 minutes late, so please adjust your time accordingly. We will start back here at approximately 1:30, and that will be just a little less than an hour for our virtual attendees to join back with us. And for those students that will be having lunch, that again is in the Inasmuch Foundation Patrons Lounge on the second floor. You can take the elevator near the registration desk to the second level. So again, thank you all for joining us. Thank you. It is our indeed extreme pleasure to have you. Such inspiring words, great, tremendous research, and I hope you can join us again soon in Stillwater. So,
thank you.
Thank you.
Thanks a lot.
Thank you.